Right now, as you settle in and the day starts to fall away, something is happening inside your skull that you have absolutely no say in. Your pupils are adjusting to the darkness. Your heart rate is slowing down beat by beat. The muscles along your jaw and your shoulders are beginning to release tension you didn't even know you were carrying. Somewhere deep in your brainstem, a cluster of cells the size of a grain of rice is deciding how often you breathe, and it has been doing this every single moment of your life without once asking for your permission. You didn't tell your stomach to start digesting whatever you ate tonight. You didn't instruct the tiny muscles around your blood vessels to relax, which is lowering your blood pressure right now as your body prepares itself for sleep. All of this is happening on its own. Or rather, all of this is being handled by a version of you that you have never met. A version that has been running the show since before you were born. So here is something worth thinking about as you lie there tonight. In September of 1848, a 25-year-old railroad foreman in Vermont named Phineas Gage was packing explosive powder into a hole in solid rock. It was routine work. He had done it hundreds of times, but on this particular afternoon, his iron tamping rod struck the rock at the wrong angle and the powder ignited. The explosion launched that rod, which was about 3.5 feet long and weighed more than 13 pounds, straight through his left cheekbone, up through the front of his brain and out the top of his skull. And here is the part that makes this story unlike almost anything else in the history of medicine. Phineas Gage survived. He was conscious within minutes. He spoke. He walked. By all standard measures, he was fine. He could see, he could hear, he could move, he could remember his past. His intelligence seemed intact. His language was perfectly normal. But in the weeks and months that followed, the people who knew Gage began to say something that no one quite knew how to explain. They said he was no longer Phineas Gage. The man who had been polite, responsible, well-liked by everyone on his crew, had become impulsive, crude, and unreliable. He cursed constantly. He made plans and abandoned them within minutes. He couldn't hold down a job. His doctor later wrote that the balance between his intellectual faculties and his animal propensities had been destroyed. The iron rod had not taken away his ability to think, or speak, or move. It had taken away something far more unsettling. It had taken away who he was. And that fact, when you really stop and sit with it, opens up a series of questions that are genuinely strange. Because if a metal rod through a particular patch of brain tissue can change your personality, your morality, your sense of responsibility, then those things were never floating around in some abstract soul or spirit. They were, the whole time, living in a specific physical location inside your head. They were made of cells, and cells can be damaged, which leads to the questions we are going to sit with tonight, slowly, one by one, in no particular rush. And these are not abstract questions. They are about you, about what is happening inside your skull right now, in this room, in this bed, as you listen to these words. Your brain contains two hemispheres, connected by a bridge of nerve fibers. If that bridge is cut, something happens that sounds impossible, but has been documented dozens of times. Two separate minds appear inside the same skull, each with its own perception, its own preferences, its own will. So the first question is this. Right now, as you lie here feeling like one person, how many of you are actually in there? The second question. Your heart is beating right now without your permission. Your stomach is digesting without your input. Your pupils adjusted to this room without you telling them to. How much of the rest of what you do, the decisions you make, the things you say, the person you think you are choosing to be, is also running on its own, handled by brain systems you have never met and will never meet? And then the deepest question of all. Right now, you feel like a single person. You feel like there is one continuous you who woke up this morning, who lived through this day, who is lying here now. That feeling of unity is the most familiar thing in your life. But what if it is a construction? What if the sense of being a single, unified author of your own story is something your brain assembles, moment by moment, from the work of dozens of independent systems? And what if the assembly is so seamless that you never notice the seams? We are going to walk through the entire story tonight. We will start thousands of years ago, with the first people who ever tried to figure out where the mind lives. We will work our way through the anatomy, piece by piece, slowly, so that by the time we get to the strange experiments and the startling case studies, you will understand exactly why they matter. And along the way, we will meet the people who cracked these questions open, one after another, each discovery leading to the next, each answer revealing a deeper mystery underneath. There is no rush at all. If you drift off somewhere along the way, that is perfectly fine. The story will still be here. So just settle in. Let your breathing find its own rhythm, and let's begin at the very beginning. For most of human history, nobody was sure where the mind actually lived. And the debate was not casual. Some of the greatest thinkers who ever lived got this question completely wrong, and they got it wrong for reasons that made perfect sense at the time. Start with Aristotle, 4th century BC, one of the most brilliant people in the history of the world.
Aristotle believed that the heart was the seat of thought, emotion, and intelligence. And if you had been standing in ancient Athens, watching him make his case, you might have believed him. Because think about what the heart does when you experience something emotionally significant. Your pulse races when you are afraid. Your chest aches when you grieve. Your heart seems to respond to the world in a way that the brain simply does not. Nobody has ever clutched their skull in a moment of heartbreak. The heart feels like it is where experience happens. And then there was the brain itself. When Aristotle looked at it, he saw a cool, wet, greyish mass that did not seem to do much of anything. It did not pulse. It did not respond obviously to stimulation. His best guess was that the brain existed to cool the blood, like a kind of radiator for the body. A sophisticated piece of plumbing, but not the seat of the soul. Here is the thing. Aristotle was not some fool who missed the obvious answer. The brain gives almost no outward indication of what it does. It does not move. If you poke it, the person does not feel pain because the brain has no pain receptors. It just sits there, pale and wrinkled and silent, offering no clues. So Aristotle looked at the evidence available to him, and he made a reasonable inference. He was wrong, but he was wrong in the way that smart people are often wrong, by trusting what was visible and missing what was hidden. Not everyone agreed with him, even at the time. Hippocrates, who lived about a generation before Aristotle, had a very different view. Hippocrates had spent years treating patients with head injuries, and he had noticed something that pointed in a completely different direction. People who took blows to the left side of the head sometimes lost the ability to move the right side of their body. People with damage to the back of the skull sometimes lost their vision. These patterns were too consistent to be coincidental. And so Hippocrates wrote something remarkably modern for his era. He wrote that from the brain, and from the brain only, arise our pleasures, joys, laughter, and sorrows, our pains, our griefs, our tears. He placed everything, all of human experience, inside the skull. But Hippocrates could not explain how. He could not point to a mechanism. He had observation without theory. And observation without theory tends to lose arguments to elegant wrong theories. Aristotle's framework was more complete, more logically satisfying, and so for centuries the heart continued to be the default answer in much of the Western world. You can still hear the echoes of this in everyday language. We say, learn it by heart.
We say someone is heartless when they lack empathy. We talk about heartfelt emotions. The vocabulary of feeling is still mapped onto the chest, 2,000 years after Aristotle. The person who started to shift the balance was a Roman physician named Galen, working in the 2nd century AD. Galen had an unusual laboratory. He was, among other things, the doctor for the gladiators of Pergamon, and gladiators sustained every kind of injury imaginable. Galen saw men with crushed skulls who could no longer speak. He saw men with wounds to specific parts of the brain who lost specific abilities while retaining others. He also did extensive work with animals, carefully severing nerves in living pigs to see what happened when specific connections to the brain were cut. And what he found, again and again, was that the brain was in charge. Cut a nerve leading from the brain to the larynx, and the pig fell silent. Damage the back of the brain, and movement was affected. The evidence was overwhelming, but Galen added his own theory on top of the evidence, and the theory, as it turned out, would hold medicine back for more than 1,000 years. He believed that the brain worked through what he called animal spirits, a kind of fluid that was manufactured in the hollow cavities inside the brain, what we now call the ventricles, and then pumped out through the nerves like water through pipes. The nerves, in Galen's model, were hollow tubes. The muscles inflated when these spirits flowed into them, like tiny balloons. It was a hydraulic theory of the mind. And you can see why someone might believe that. If you cut a nerve, the muscle it supplies goes limp. It looks like something stopped flowing through the tube. The ventricles really are filled with fluid, cerebrospinal fluid, and they are obvious when you dissect a brain. Everything fit. So Galen's model, a brain that works by pumping fluid through hollow nerves, became the dominant theory of how the nervous system operated. It survived through the fall of Rome, through the Middle Ages, through century after century of medicine, essentially unchallenged. It took until the 1500s for someone to begin pulling it apart. Andreas Vesalius, a young anatomist working in Padua, did something that had been surprisingly rare in the history of medicine up to that point. He actually looked. He dissected human bodies with extraordinary care, and he drew what he found with a precision that had never been attempted before. His great book, published in 1543, contained illustrations of the brain that were accurate enough to be recognizable to a modern neuroanatomist. And one of the things Vesalius noticed was that the ventricles, those fluid-filled cavities that Galen had made the centerpiece of his theory, were not particularly special. Other animals had them too. A sheep's ventricles looked a lot like a human's. If the ventricles were where intelligence lived, then sheep should be writing poetry. Vesalius did not have a replacement theory. He could see that Galen's model was wrong, but he could not yet explain what the brain was actually doing. That would take another 300 years of work, one discovery building on the next, each step making the picture slightly clearer. And one of the first and most dramatic of those steps began on a September afternoon in 1848, on a railroad construction site in the hills of Vermont, when a tamping iron punched through the skull of a man named Phineas Gage and changed the way we think about who we are. Now, I want to linger on Gage for a moment longer, because his case was not just a medical curiosity, it was a philosophical earthquake, and the aftershocks are still being felt today. Before Gage, most educated people in the Western world believed in something like a soul, a unitary, indivisible essence that constituted the true self. The body could be damaged, limbs could be lost, organs could fail, but the essential person, the soul, was presumed to be immune to physical injury. Gage's case attacked this assumption at its foundation, because if a metal rod through the front of the brain could change your personality, your values, your sense of responsibility, then those qualities were not properties of an immaterial soul. They were properties of brain tissue. They were physical, material, and therefore destructible. The physicians who examined Gage initially struggled to make sense of what had happened. Some doubted the reports, arguing that personality changes after brain injury were impossible, or at least greatly exaggerated. Others accepted the reports but drew limited conclusions, noting only that the frontal lobes seemed to be involved in higher functions, without specifying what that meant. The person who made the most ambitious attempt to localize mental functions in the brain, even before Gage, was a Viennese physician named Franz Joseph Gall. In the early 1800s, Gall proposed a system he called organology, which later became known as phrenology. Gall's idea was that different mental faculties, things like language, courage, amativeness, secretiveness, were each located in a specific organ of the brain, and that the size of each organ determined the strength of the corresponding faculty. Since the skull, he argued, conforms to the shape of the brain underneath, you could determine a person's mental characteristics by feeling the bumps on their head. Phrenology was enormously popular for several decades. People lined up to have their heads read by traveling phrenologists. Employers used it to screen job applicants. Courts considered it as evidence. For a while, it seemed like the answer to the ancient question of how the mind was organized. And then it fell apart. Not because the basic idea of localization was wrong, but because Gall's specific claims could not be verified. No one could find consistent correlations between skull bumps and personality traits. The faculties Gall proposed were arbitrary and unsupported by evidence. His methods were unscientific, relying on anecdotes and confirmatory cases rather than controlled studies. By the middle of the 19th century, phrenology had been thoroughly discredited, and it dragged the entire concept of functional localization down with it. This created an unfortunate overcorrection. For a generation after phrenology's collapse, many scientists swung to the opposite extreme and argued that the brain was equipotential, meaning that all parts of the brain contributed equally to all mental functions and that no specific region had a specific job. According to this view, the brain was like a bowl of porridge, and the effect of damage depended only on how much brain tissue was lost, not on which tissue was lost. Gage's case, and then Broca's discovery, pulled the pendulum back. The brain was not porridge, it had structure. Different parts really did do different things. But the truth was more nuanced than either the phrenologists or the equipotentialists had imagined. Some brain functions are sharply localized, handled by a small, specific region. Others are distributed across large networks of regions working together. And many functions involve both localized components and distributed networks simultaneously. Modern neuroscience, using tools like functional MRI that can image brain activity in real time, has revealed a picture that is both structured and complex. There are regions that respond very specifically to certain kinds of stimuli. Regions that light up when you see faces, but not when you see objects. Regions that respond to places, but not to faces. Regions dedicated to processing language. Regions dedicated to understanding other people's mental states. But these regions do not work in isolation. They are constantly communicating with each other through those white matter highways, forming networks that shift and reconfigure depending on the task. The brain has both geography and connectivity. It is a landscape of specialized regions linked by information highways, and the mind emerges from the interplay between the two. Understanding this interplay is, arguably, the central challenge of 21st century neuroscience. But in 1848, none of this was known. What was known was just this. A man had a hole in the front of his brain, and the man who walked away from the accident was not the same person who had been there before. The person had changed, and the brain had changed, and the two facts seemed to be connected. So let that story settle for a moment. We have covered a lot of ground already, thousands of years of it, and we are just getting started. The ancient world gave us a debate, heart versus brain, and the brain eventually won. But winning the debate only opened a much harder question. If the mind really does live in the brain, then how is it organized in there? Is the brain a single, uniform thing, like a bowl of porridge where every spoonful is the same? Or does it have structure, with different parts handling different jobs? The story of Phineas Gage was the first powerful hint that the answer was structure. Because Gage did not lose everything when that rod went through his head. He lost something very specific. He lost the part of himself that made him a responsible, socially appropriate human being, while keeping his memory, his language, his motor skills, his intelligence by almost any measurable standard. Something up there in the front of his brain was specifically in charge of making Phineas Gage who Phineas Gage was. And when it was destroyed, everything else kept working, but the person was gone. That specificity is the key to everything that follows. Because once you realize that different parts of the brain do different things, a whole cascade of questions opens up. Which parts? What things? And what happens at the boundaries, where one region's job ends and another's begins? To answer those questions, though, we need to understand how the brain is actually built. We need to go inside the skull and look at the physical structure, the anatomy, of this 3-pound organ that somehow produces everything you have ever thought, felt, remembered, or dreamed. And that is where we are going next. But let me take it slowly, because the anatomy matters. Every piece we lay down now is going to come back later in the story, when we start talking about split-brain patients and sleepwalkers and people who lose their sense of being in control. The anatomy is not just naming parts. The anatomy is the map, and without the map, the rest of the journey does not make sense. So let's go inside. Your brain started out as almost nothing. About 2 weeks after conception, when the embryo that would become you was still smaller than a grain of rice, a flat sheet of cells on its surface began to fold inward, like a crease forming in a piece of fabric. That crease deepened, and the edges rose up and curled toward each other, and eventually they fused together to form a hollow tube. This is called the neural tube, and it is one of the most important structures in all of embryology, because everything in your central nervous system, your entire brain, your entire spinal cord, developed from this one simple tube. By the 3rd week, the front end of the tube had started to swell. 3 little bulges appeared, like beads on a string. The medical terms are prosencephalon, mesencephalon, and rhombencephalon. But what you really need to know is just this: front, middle, and back. 3 pouches. That was it. That was the beginning of a human brain. By 6 weeks, those 3 pouches had started to subdivide. The front pouch split into 2 parts. One would become the diencephalon, a deep central structure that we will talk about in a moment. The other, the telencephalon, started growing outward and expanding to the sides. If you imagine the letter T, the diencephalon is the vertical stroke, deep in the center, and the telencephalon is the horizontal bar, spreading out on both sides. That horizontal bar, as the weeks passed, kept growing and growing and folding, and by the time a baby is born, it has become the cerebrum, the enormous, wrinkled mass that takes up most of the space inside your skull. The part of the brain that most people picture when they hear the word brain. The part that, as we will see tonight, contains everything you call yourself. The back pouch subdivided too. It gave rise to structures called the medulla oblongata, the pons, and the cerebellum. The medulla and pons together form the lower part of what is called the brainstem, the oldest and most fundamental part of your brain, the part that keeps you alive in the most basic sense. Breathing, heart rate, blood pressure, swallowing, wakefulness and sleep. If the cerebrum is where you do your thinking, the brainstem is where you do your surviving. And between them, the middle pouch became the mesencephalon, or the midbrain. It is the smallest section of the brain by far, only about 3/4 of an inch in an adult, but it handles some functions that are surprisingly important, including the movement of your eyes, the detection of new sounds and new visual stimuli, and the relay of signals between the lower and upper parts of the brain. So by the time you were born, that simple hollow tube had become this: a brainstem at the base, handling the essentials; a cerebellum tucked behind and below, handling coordination and motor learning; a diencephalon in the center, acting as a relay station and hormone command center; and draped over all of it, taking up roughly 80% of the total volume, the cerebrum. Think of it as a building. The brainstem is the basement. The plumbing, the heating, the electrical grid, the things that keep the building functional. The diencephalon is the front desk, sorting the mail, directing visitors to the right floor. And the cerebrum is every floor above that: the offices, the conference rooms, the libraries, the art studios, the places where the interesting work happens. Now, I mentioned that the brainstem handles survival. Let me stay with that for a moment, because the brainstem deserves more attention than it usually gets. When most people think about the brain, they think about the cerebrum, the wrinkled outer shell, the part that does the thinking and planning and remembering. But the brainstem is, in some ways, the more important structure, because without it, there is no thinking and planning and remembering. Without it, there is no life at all. The brainstem is made up of three main parts, stacked on top of each other. At the bottom, connecting directly to the spinal cord, is the medulla oblongata. Above that is the pons. And above that is the midbrain, the mesencephalon. Together, these structures are only about 3 inches long, roughly the length of your thumb, but they contain an extraordinary density of vital circuitry. Inside the medulla, there are clusters of neurons that function as the respiratory pacemaker. These cells fire rhythmically, about once every 5 seconds in a resting adult, and each burst of firing triggers a breath. The signal travels down the spinal cord to the diaphragm and the intercostal muscles, the muscles between your ribs, and your lungs expand. As the lungs stretch, sensory receptors send a signal back up to the brainstem, reporting that the chest has expanded enough. This feedback signal terminates the inhalation and triggers the exhalation. Then a pause. Then the pacemaker fires again, and the cycle repeats. Every breath you have ever taken in your entire life was initiated by this tiny cluster of cells. And the system runs automatically. You do not have to think about breathing, although you can choose to override the system temporarily, holding your breath or breathing faster. But when you stop paying attention, the pacemaker takes over again without any gap. Right next to the respiratory center sits the cardiovascular center, which regulates heart rate and blood vessel tone. When the concentration of carbon dioxide in your blood rises, both centers respond together. Your breathing speeds up and your heart beats faster, pumping more blood through the lungs, picking up fresh oxygen and offloading the excess carbon dioxide. These two systems are coupled because they solve the same fundamental problem: keeping your tissues supplied with oxygen. The brainstem also houses the nuclei of most of the cranial nerves, the 12 pairs of nerves that connect your brain directly to structures in your head and body without going through the spinal cord. And each of these cranial nerves has its own personality, its own function, its own territory. The fifth cranial nerve, the trigeminal, is the main nerve for facial sensation. It has 3 branches, one for the forehead, one for the upper jaw, and one for the lower jaw, and it carries touch, pain, and temperature information from the entire face. When your teeth ache, that signal is traveling through the trigeminal nerve. The 7th nerve, the facial nerve, controls the muscles that produce facial expressions. The smile you give a friend, the frown you make when you are concentrating, the raised eyebrows of surprise, all of these movements are coordinated by the facial nerve. But the facial nerve does more than just move muscles. It also carries taste information from the front 2/3 of the tongue, and it controls the lacrimal glands, the glands that produce tears. So when you cry, that is the facial nerve at work. The eighth nerve, the vestibulocochlear nerve, comes from the inner ear and splits into two branches. One branch carries hearing signals from the cochlea, the snail-shaped organ that converts sound waves into electrical impulses. The other branch carries balance signals from the vestibular apparatus, the set of fluid-filled canals and chambers that tell your brain which way is up and how your head is moving. And then there is the 10th cranial nerve, the vagus nerve, which we have already met. The vagus is the longest of the cranial nerves and arguably the most important. Its name means wandering in Latin, and the anatomists who first described it were amazed by how far it travelled. It exits the brainstem, passes through the neck alongside the esophagus, enters the chest, and sends branches to the heart, the lungs, and the bronchial tubes, passes through the diaphragm, and then fans out across the stomach, the intestines, the liver, and the other abdominal organs.
The vagus is the main channel through which the parasympathetic nervous system communicates with the internal organs. When your heart slows as you relax, that is vagal signaling. When your stomach churns after a meal, vagal fibers are coordinating the contractions. When you hiccup after eating too fast, that is a vagal reflex triggered by gastric distension. There is a detail here that connects beautifully to our story about the stranger. The vagus nerve also carries sensory information from the internal organs back up to the brain. It reports on the state of your gut, the pressure in your stomach, the chemical environment of your intestines. And some researchers believe that these vagal signals contribute to what we colloquially call gut feelings. Those vague but sometimes powerful intuitions that something is right or wrong, safe or dangerous. Your gut is, in a literal sense, talking to your brain.
And the brain is listening, even though you rarely become conscious of the conversation. All of these brainstem functions, the breathing, the heartbeat, the cranial nerve reflexes, the vagal regulation of internal organs, they are ancient. They evolved hundreds of millions of years ago in the earliest vertebrates, long before the cerebrum existed. A fish has a brainstem. A frog has a brainstem. A lizard has a brainstem. The circuitry that keeps you alive tonight is fundamentally the same circuitry that kept a Devonian fish alive 400 million years ago. Evolution did not need to reinvent the basics, it just kept adding layers on top. And this is, in a way, the architectural principle of the entire brain. The newest structures sit on top of the oldest. The cerebral cortex, the most recent evolutionary addition, wraps around the outside, handling the most complex and most recently evolved functions: language, abstract thought, planning, moral reasoning. The brainstem, the oldest part, sits at the core, handling the most fundamental and most conserved functions: breathing, heart rate, wakefulness, basic reflexes. And in between, the diencephalon and the limbic system handle functions that are somewhere in the middle: emotion, motivation, memory, hormonal regulation. Your brain is a geological record of evolution. The deepest layers are the oldest, the surface is the newest, and the stranger, the unconscious machinery that runs your body, lives mostly in the deep layers, in the ancient structures that have been doing their job since before your ancestors crawled out of the sea. One detail about brain development that connects to everything we have been discussing is the process of pruning. A newborn baby has, proportionally, far more neurons than it needs. The infant brain is like an overgrown garden, dense with connections, many of which are redundant or disorganized. In the first few years of life, a massive program of elimination takes place. Synapses that are used are strengthened. Synapses that are not used are eliminated. Entire neurons that do not find a useful role in the developing circuits are allowed to die. This might sound wasteful or even alarming, but it is actually the mechanism by which experience sculpts the brain. A newborn brain is prepared for any environment. It has the raw wiring to learn any language, adapt to any culture, respond to any set of sensory inputs. But as the child grows and encounters a specific environment, with specific sounds, specific visual patterns, specific social interactions, the brain prunes away the connections that are not needed and strengthens the ones that are. A Japanese baby hears Japanese and strengthens the circuits for processing Japanese phonemes while pruning the circuits for phonemes that do not occur in Japanese. An English-speaking baby does the opposite. By the end of the first year, a baby's auditory cortex has been customized for its linguistic environment, and the ability to distinguish certain foreign phonemes has been significantly reduced. This is why early experience matters so much. The pruning process creates a brain that is exquisitely adapted to its specific environment, but the adaptation comes at a cost. The eliminated connections cannot easily be rebuilt later. The critical periods, those windows of opportunity during which the brain is maximally plastic and responsive to environmental input, eventually close. And once they close, certain kinds of learning become much harder, though not impossible. It is a poignant bargain. You get a brain that is perfectly tuned to your world, but you lose the capacity to have been perfectly tuned to a different world. The stranger that you carry with you tonight was shaped, in its deepest structure, by the world you encountered as a child. The sounds you heard, the faces you saw, the hands that held you, the languages that surrounded you. All of these left their mark in the pattern of your synaptic connections, in the architecture of your cortex, in the stranger that now runs your body while you sleep. Now, if you were to take the cerebrum and cut it in half from front to back, right down the middle, you would find that it comes apart into two halves. These are called the left and right hemispheres, and they are connected by a thick bridge of nerve fibers called the corpus callosum. Remember that name, because the corpus callosum is going to become very important later in our story, when we talk about what happens when it is cut. The corpus callosum contains somewhere around 200 million nerve fibers, and its job is to allow the left hemisphere and the right hemisphere to talk to each other, to share information, to coordinate. Every time you do something that requires both sides of your brain, which is almost everything, those signals are racing back and forth through the corpus callosum. Each hemisphere is divided into 4 lobes, and each lobe has at least one major specialty. The frontal lobe is at the front, obviously, and it handles planning, decision-making, voluntary movement, and something that is harder to pin down, but that we might call personality. The parts that make you act like a responsible social being. The parts that allow you to weigh consequences before you act. Those live in the frontal lobe, especially in the very front part, called the prefrontal cortex. And if this sounds familiar, it should, because the prefrontal cortex is exactly the region that was destroyed in Phineas Gage. Behind the frontal lobe, separated by a deep groove called the central sulcus, is the parietal lobe. This is where your brain processes touch, pressure, temperature, the position of your body in space, that sense you have of where your limbs are even with your eyes closed. If you reached out in the dark right now and touched your nose with your fingertip, you could do it easily. And the reason you could do it is that your parietal lobe is constantly updating a map of where every part of your body is. Down on the sides, roughly behind your ears, are the temporal lobes. These handle hearing, the processing of language, and a significant chunk of memory, including the hippocampus, a seahorse-shaped structure buried deep inside the temporal lobe that is critical for forming new memories. We will come back to the hippocampus when we talk about a patient named Henry Molaison, whose story will change the way you think about what memory actually is. And at the very back, tucked into the rear of the skull, is the occipital lobe. Vision. Everything you see is processed here.
The light entering your eyes right now, even in this dim room, is being turned into electrical signals that travel through the optic nerve, pass through the thalamus, and arrive in the occipital lobe, where specialized neurons begin the work of figuring out what you are looking at. Now, here is something about the anatomy that matters enormously for our story tonight. The outer surface of the cerebrum, the thin layer that covers the entire thing like bark on a tree, is called the cerebral cortex. It is only about 1/8 of an inch thick in most places, but it contains roughly 16 billion neurons. And this is where the highest level processing happens. Consciousness, language, abstract thought, moral reasoning, the sense of being a person. All of it lives in this thin rind of grey matter on the outside of the brain. And when I say grey matter, that is not just a figure of speech. If you were to look at a cross-section of the brain, you would see two distinct colours. The outer layer is greyish, and the tissue deeper inside is whitish. The grey matter is where the cell bodies of neurons are concentrated, where the actual computing happens, where signals are received and processed, and decisions are made. Think of it like a room full of people all talking to each other, huddling together, processing information. The white matter underneath is made up of long nerve fibers called axons, coated in a fatty insulation called myelin. And these are the cables that connect one region to another. The white color comes from the myelin. So grey matter is where the thinking happens, and white matter is how different thinking regions talk to each other. Deep inside the brain, below the cortex and the white matter, there are also clusters of grey matter called nuclei. These are little islands of cell bodies surrounded by white matter, and they handle specific tasks. The basal nuclei, for example, which sit deep in the cerebrum, are involved in coordinating learned movements, the kind of smooth, automatic motion that you do without thinking about it. Walking, reaching for a cup. The movements that feel effortless precisely because the basal nuclei have practiced them so many times that they run on autopilot. And the limbic system, a group of structures that includes the amygdala and the hippocampus, sits deep in the temporal lobe and along the inner surface of the hemispheres. The limbic system handles emotion, motivation, memory formation, and the connection between what you feel and what you do. The amygdala, which is roughly the size and shape of an almond, is your brain's alarm system. It evaluates incoming information for potential threats and triggers the fight or flight response when it detects danger. If you have ever stepped off a curb and seen a car coming from the corner of your eye and jumped back before you even consciously registered what was happening, that was your amygdala, working faster than your conscious mind could follow. We are moving slowly here on purpose, because every one of these structures is going to come back into the story. The prefrontal cortex will matter when we talk about creativity and letting go of conscious control. The corpus callosum will matter when we talk about split brain patients. The hippocampus will matter when we talk about memory and identity. The thalamus will matter when we talk about consciousness and anesthesia. All of these pieces connect. So if you are still with me, that is wonderful. And if you are starting to drift, that is fine too. There is no test at the end of this. Just let the information settle, and we will keep going. Because now that we have the map of the brain, we need to understand the language that the brain speaks. How do those billions of neurons actually communicate with each other? What is a thought at the physical level? What is happening right now inside your head as you listen to these words? The answer involves electricity, chemistry, and one of the most elegant signaling systems in all of biology.
and the story of how we figured it out involves a dead frog, a thunderstorm, and a rivalry between two Italian scientists that lasted for decades. In the late 1700s, an Italian physician named Luigi Galvani was conducting experiments in his laboratory in Bologna. He had a dead frog laid out on his workbench, its legs detached from its body, and he was studying the animal's muscles. During a thunderstorm, as the story goes, Galvani noticed that when he touched the frog's leg with a metal instrument, while lightning flickered in the distance, the leg twitched. The muscle contracted as if the frog was still alive. Galvani was fascinated. He repeated the experiment under different conditions, and he concluded that the frog's tissues contained a form of electricity, what he called animal electricity. He believed that living things generated their own electrical charge, and that this electricity was what made muscles move and nerves carry signals. Another Italian scientist, Alessandro Volta, disagreed. Volta thought the electricity was not coming from the frog at all, but from the contact between two different metals used in the experiment. He argued that Galvani was seeing a chemical reaction, not a biological phenomenon. And to prove his point, Volta built a device that could generate electricity without any animal tissue whatsoever. A stack of alternating zinc and copper discs, separated by pieces of cardboard soaked in salt water. This was the first battery, and Volta was right that the metals were generating a current. But here is the thing. Galvani was also right. Both men were correct about different aspects of the phenomenon. The metals were indeed producing electricity through a chemical reaction, just as Volta said. But living tissues also carry electrical signals, just as Galvani had observed. It took decades for this to be sorted out, and the person who really resolved the question, who proved beyond any doubt that nerves carry electrical impulses, was a German physiologist named Émile Dubois-Reymond, working in the 1840s. Dubois-Reymond built sensitive instruments that could detect the tiny electrical signals flowing through nerve fibers, and he measured what he called the negative variation, which was not the catchiest name, but what he was describing was nothing less than the nerve impulse itself. The fundamental unit of communication in the nervous system. So what exactly is a nerve impulse? How does a neuron actually send a signal? Every neuron in your body has the same basic structure.
There is a cell body called the soma, which contains the nucleus and all the standard cellular machinery. Branching out from the soma are a set of short, tree-like extensions called dendrites. These are the receiving end of the neuron. They detect stimuli, whether that is a chemical signal from another neuron or a physical touch on your skin, or light hitting the back of your eye. And extending away from the soma in the other direction is a single long fiber called the axon. This is the transmission line. This is how the neuron sends its signal from one place to another. The key to understanding how a neuron fires is voltage. Every neuron, when it is at rest, carries a slight negative charge on the inside of its membrane compared to the outside, roughly negative 70 millivolts. This is called the resting potential, and it exists because of the way ions, which are atoms with an electrical charge, are distributed across the cell membrane.
There is a lot of sodium outside the cell and a lot of potassium inside the cell, and the membrane has special proteins called ion channels that control whether these ions can flow in or out. When a stimulus reaches the dendrites, something called a sodium channel opens. Sodium ions, which carry a positive charge and which are much more concentrated outside the cell, rush in through the open channel. This makes the inside of the neuron a little less negative, a little more positive. And if enough sodium flows in to push the voltage up to a critical threshold, roughly negative 55 millivolts, something dramatic happens. Hundreds of additional sodium channels, called voltage-gated sodium channels, snap open all at once. Sodium floods into the cell, the voltage spikes upward past 0 to around positive 30 or 40 millivolts, and the neuron fires. This spike is called an action potential. The whole event takes about 1 millisecond, 1/1000th of a second. And it is an all-or-nothing event. Either the threshold is reached and the neuron fires completely, or it is not reached and nothing happens. There is no such thing as a half-hearted action potential. Once the action potential is generated at the cell body, all that positive charge rushes down the axon like a wave traveling along a rope. And when it reaches the very end of the axon, the terminal, it triggers the release of chemical messengers called neurotransmitters. These chemicals are stored in tiny bubbles called synaptic vesicles, and when the action potential arrives, calcium ions flow into the terminal and push those vesicles toward the membrane, where they burst open and spill their contents into a tiny gap between the end of the axon and the next cell. That gap is about 20 nanometers wide, which is roughly 1,000 times thinner than a human hair. This gap is called the synapse. The neurotransmitters drift across the synapse, bind to receptors on the surface of the next cell, and depending on the type of neurotransmitter, they either encourage that next cell to fire or discourage it from firing. Excitatory neurotransmitters like glutamate push the next neuron toward firing. Inhibitory neurotransmitters like GABA pull it away from firing. The decision about whether any given neuron will fire depends on the sum of all the excitatory and inhibitory signals it is receiving from thousands of other neurons at the same time. And every neuron releases only one type of neurotransmitter. A neuron that uses serotonin will always use serotonin. One that uses dopamine will always use dopamine. The identity of a neuron is partly defined by which chemical it speaks. Think about that for a moment. Every thought you have, every sensation you feel, every memory you recall, every decision you make is produced by billions of these tiny electrical spikes and chemical releases happening in patterns across your brain. Right now, as you listen to these words, specific networks of neurons are firing in your auditory cortex, processing the sounds. Other networks in your language centers are extracting meaning from those sounds. Still other networks in your frontal lobes are following the thread of the argument, connecting what I am saying now to what I said a few minutes ago. And all of it is running on electricity and chemistry, on sodium and potassium and calcium, on glutamate and GABA and dopamine. One more detail about the anatomy of signaling, and then we will move on. Many of the longest axons in the brain are wrapped in that fatty insulation I mentioned earlier, myelin. Myelin is produced by special cells called oligodendrocytes inside the central nervous system and by different cells called Schwann cells in the peripheral nervous system. The myelin wraps around the axon in segments, with small gaps between each segment, and the action potential jumps from gap to gap rather than flowing continuously along the entire length. This makes the signal travel much faster. In unmyelinated fibers, signals travel at maybe 3 to 6 feet per second. In myelinated fibers, the speed can reach over 250 miles per hour. Still not as fast as electricity in a copper wire, not even close, but fast enough to allow your brain to coordinate the movements of your body in real time. And the white matter we talked about earlier? That whiteness comes from the myelin. The white matter of the brain is essentially a vast network of myelinated axons, cables carrying information from one region to another, connecting the brainstem to the cortex, the left hemisphere to the right, the front of the brain to the back. The brain's internal internet. So that is how neurons talk: electrical spikes, chemical handoffs, insulated cables. A language that was completely invisible to every physician and philosopher who ever lived, until a handful of scientists in the 18th and 19th centuries started to crack the code. Now, here is why all of this matters for our story tonight. Once scientists understood that the brain communicated through electrical signals traveling along specific pathways, they could start asking a much more interesting question. If the brain is made of billions of neurons connected in specific patterns, and if damage to specific patterns causes specific problems, then maybe, just maybe, the brain is not a single, undifferentiated organ. Maybe it has regions, departments, areas of specialization. And maybe the reason Phineas Gage lost his personality but not his language was that personality and language live in different neighborhoods. That idea, the idea that specific mental functions are handled by specific parts of the brain, is called functional localization. And the next major piece of evidence for it came not from a railroad accident, but from a hospital in Paris in 1861, when a surgeon named Paul Broca met a patient who could only say one word. The patient's name was Louis Victor Le Bon, but everyone at the hospital called him Tan. They called him Tan because that was the only syllable he could produce. When you asked him a question, he would say, "Tan, tan." When he was frustrated, he would say, "Tan, tan, tan." He could understand language perfectly well. If you spoke to him, he clearly comprehended what you were saying. He could follow instructions. He would nod or shake his head appropriately. His intelligence seemed largely intact. But he could not speak. He could not produce words. The only sound he could make was that single, repeated syllable. Paul Broca was a surgeon and an anthropologist, and he was deeply interested in the question of whether different parts of the brain served different functions. When Le Bon died, Broca performed an autopsy and found that there was a large lesion, an area of dead tissue, in the left frontal lobe of his brain. Specifically, in the lower part of the left frontal lobe, in a region just forward of the motor cortex. Broca presented this case to the Anthropological Society of Paris, and then he found more cases. Over the next few years, he identified several more patients who had lost the ability to produce speech, and in every case, the damage was in the same general area of the left frontal lobe. This region became known as Broca's area, and the condition, the inability to produce speech despite intact comprehension, became known as Broca's aphasia. Broca's famous conclusion, which he announced in 1865, was that we speak with the left hemisphere. Language production, he argued, was localized to a specific region of the left frontal lobe. About a decade later, a German neurologist named Carl Wernicke found the other side of the puzzle. Wernicke described patients who had the opposite problem. They could speak fluently. Words poured out of them in grammatically correct sentences, but the words did not make sense. They would string together phrases that sounded like language but communicated nothing. And more importantly, they could not understand language when it was spoken to them. If you said, "Sit down," they would stare at you blankly. They heard the sounds, but the sounds had lost their meaning. Wernicke found that these patients had damage in a different location, in the upper part of the left temporal lobe, near the junction with the parietal lobe. This region became known as Wernicke's area, and the condition became known as Wernicke's aphasia. So now there were two language regions, and they did different things. Broca's area handled the production of speech, the motor coordination and planning required to turn thoughts into words. Wernicke's area handled the comprehension of speech, the ability to extract meaning from the sounds you hear. And the two regions were connected by a bundle of nerve fibers, allowing them to work together. You hear a question. Wernicke's area processes the meaning. The meaning is sent forward to Broca's area, and Broca's area coordinates the muscles of your mouth and throat and tongue to produce an answer. And notice something important. Both regions were in the left hemisphere. For most people, language is heavily lateralized to the left side of the brain. The right hemisphere has some role in language, particularly in understanding tone of voice and emotional nuance. But the core machinery of language, the ability to produce and comprehend words, is predominantly a left hemisphere function. This is remarkable if you think about it. It means the two halves of your brain are not mirror images of each other. They do not do the same things. The left hemisphere, in most people, handles language, logical reasoning, and detailed analytical processing. The right hemisphere handles spatial awareness, the recognition of faces and emotions, the processing of music, and a kind of holistic pattern recognition that works differently from the left hemisphere's more sequential approach. You are, in a sense, already two. Your brain has two halves, and those halves have different specialties. They work together seamlessly because the corpus callosum is constantly shuttling information back and forth between them. But underneath that smooth coordination, they're doing different jobs. Which raises an extraordinary question. What happens if you cut the connection? What happens if you sever the corpus callosum and prevent the two hemispheres from talking to each other? The answer to that question turned out to be one of the most astonishing findings in the entire history of neuroscience. And it began with a treatment for epilepsy. In epilepsy, a seizure is essentially an electrical storm in the brain. A group of neurons begins firing out of control, and the abnormal activity spreads, recruiting more and more neurons into the chaotic pattern. In many cases, a seizure starts on one side of the brain and then spreads to the other side by traveling through the corpus callosum, that great bridge of 200 million fibers connecting the hemispheres. Once the seizure crosses to the other side, it becomes a generalized seizure, affecting the entire brain, and the person loses consciousness, falls, and may convulse. For patients with severe epilepsy, patients whose seizures did not respond to medication and who were having dozens of seizures a day, doctors in the 1960s began performing an operation called a corpus callosotomy. They cut the corpus callosum. The idea was that if the bridge was severed, a seizure that started on one side could no longer cross to the other side, and the seizure would be contained. The surgery worked. Seizures were dramatically reduced.
And at first, the patients seemed surprisingly normal afterward. They could walk, talk, eat, carry on conversations, go about their daily lives. The two hemispheres seemed to manage just fine without the connection. Some doctors even joked that the only purpose of the corpus callosum was to transmit seizures. But a neuroscientist named Roger Sperry and his graduate student, Michael Gazzaniga, suspected that something much more interesting was happening beneath the surface of that apparent normality. They designed a series of experiments to test what each hemisphere could and could not do when it was working on its own. The key to their experiments was a simple fact about the anatomy of vision. When you look straight ahead, everything in your left visual field, everything to the left of where you are fixating, sends information only to the right hemisphere, and everything in your right visual field sends information only to the left hemisphere.
Normally, this does not matter, because the corpus callosum immediately shares the information between the two sides. But in a split-brain patient, the information stays trapped in one hemisphere. So Sperry and Gazzaniga could flash a word or an image to just one visual field, and only one hemisphere would see it. And because language is in the left hemisphere, the patient could only verbally report what the left hemisphere had seen. The right hemisphere might see something completely different, and the patient would have no conscious verbal awareness of it. The results were genuinely astonishing. In one experiment, they flashed the word Texas to the left visual field of a patient named Joe. This meant the word went exclusively to Joe's right hemisphere, the hemisphere that cannot speak. When they asked Joe what he had seen, he said he had not seen anything. His left hemisphere, which controls speech, genuinely had no idea. But when they asked Joe to draw what he had seen, using his left hand, which is controlled by the right hemisphere, Joe drew the word Texas. He drew it upside down, but he drew it perfectly legibly. And when the researchers asked him what he had drawn, he looked at his own drawing and said he did not know. He could not read it. His left hemisphere, the one doing the talking, had no access to what his right hemisphere had seen or drawn. Then they flashed the word again and asked him to draw something associated with what he had seen. Joe's left hand drew a cowboy hat. When asked what he had drawn, his verbal left hemisphere said, "A cowboy hat." And when asked if he had seen the word Texas, he said no. Think about what is happening here. Inside one skull, there are two separate centers of awareness. The right hemisphere sees a word, understands it, and can act on it.
The left hemisphere does not see the word, cannot act on it, and constructs an explanation for behavior it does not understand. Joe's left hemisphere saw his own hand drawing a cowboy hat and came up with a plausible story for why, even though the real reason was completely inaccessible to it. Gazzaniga called the left hemisphere the interpreter. He argued that one of the left hemisphere's primary jobs is to take whatever happens, whatever the body does, whatever signals come in from the world, and construct a coherent narrative to explain it. The left hemisphere is a storyteller. It needs things to make sense. And when it does not have access to the real reason for something, it simply makes one up, and it does so with complete confidence. If you drift off for a moment and come back, do not worry. We are just going to keep walking through the story. In another line of experiments, researchers tested split-brain patients with tasks that required both hands.
They showed two different shapes on the screen, one to each visual field, and asked the patient to draw both shapes simultaneously. The left visual field shape with the left hand, and the right visual field shape with the right hand. A person with an intact corpus callosum struggles with this, because the motor commands from one hemisphere interfere with the motor commands from the other. Your two hands start doing the same thing, even though you are trying to make them do different things. But split-brain patients could do it perfectly. Each hand drew its assigned shape without any interference from the other side, because the two hemispheres were no longer communicating. It was, as one researcher described it, like having two separate minds controlling the two hands. Sometimes the two hemispheres would actually conflict with each other. There are reports of split-brain patients whose left hand would reach for one shirt in the morning, while the right hand reached for a different one.
One hand would button a button, while the other unbuttoned it.
In these moments, the patient was, in a very real sense, two people. Two minds with different preferences, inhabiting the same body, unable to negotiate because the phone line between them had been cut. Michael Gazzaniga, reflecting on decades of this research, put it this way: each of us has a sense that we are a unitary being. But actually that feeling disguises the fact that each of our minds is composed of lots of different pieces that are doing different things. Different information can be represented in different parts of that machinery, and so a search for, where am I in all of this, is a little bit misguided, because the I is not such a unitary thing in the first place. That is a strange thing to sit with. The feeling of unity, the sense that there is a single you looking out through your eyes, is one of the most fundamental aspects of conscious experience. And the split-brain research suggests that this unity is, at least in part, a construction, a product of the corpus callosum doing its job so well that you never notice the seams. When the callosum is intact, the interpreter in your left hemisphere has access to everything, and so it can weave a seamless story. When the callosum is cut, the seams become visible, and you can see that the single person was, all along, a collaboration. Now, here is an important thing to say. Split-brain patients are still people. They are not two separate individuals walking around in one body. In everyday life, they function remarkably well because the two hemispheres can still share some information through other, smaller connections, and because the body itself provides a common ground. Both hemispheres see what happens when the body acts, and both can learn from the results. The cooperation is, as one researcher put it, on the outside, not on the inside.
The two minds coordinate through the shared world, even though the internal highway has been closed. There is something worth saying about what the split-brain research reveals about everyday intact brains. because the implications go well beyond the rare patients who have had their corpus callosum severed. In a normal brain, the interpreter in the left hemisphere has access to all the information from both sides, because the corpus callosum is intact. And so the stories it constructs are usually consistent with the evidence. But the key word is stories. The interpreter is not simply reporting on reality. It is constructing a narrative. It is taking a complex, messy, multi-stream flow of neural activity and turning it into a coherent account with a clear protagonist, you, and a clear sense of authorship and intention. And sometimes, even in perfectly healthy brains, the narrative diverges from reality. You have probably experienced this yourself.
You make a decision, perhaps on impulse, and then afterward, if someone asks you why you did it, you come up with a reason. The reason sounds logical, feels true, and you believe it completely. But the truth is that you did not make the decision for that reason. You made it for some other reason, perhaps an emotional impulse, a habitual response, a subconscious association. And then your interpreter constructed a plausible explanation after the fact. Social psychologists have demonstrated this in experiment after experiment. In one classic study, shoppers were shown 4 pairs of stockings and asked to choose the best one. In reality, all 4 pairs were identical, but people consistently preferred the pair on the far right, a well-documented position effect. When asked why they chose that pair, they never said, because it was on the right. Instead, they gave elaborate explanations about the fabric quality, the color, the texture.
They believed their own explanations completely, even though the real cause of their choice was something they were not aware of. This is the interpreter doing what it does best, making sense of something that, from the conscious perspective, does not have an obvious explanation, filling in the gaps, building the story. The fact that the story is wrong does not bother the interpreter at all, because the interpreter never knows it is wrong. It does not have access to the real cause. It only has access to the outcome, and it reverse engineers a plausible cause from the outcome. Gazzaniga tells a wonderful story from his split brain research that illustrates this perfectly. He flashed the word walk to the right hemisphere of a split brain patient. The patient stood up and started walking toward the door.
When Gazzaniga asked him why he was walking, the patient's left hemisphere, which had no idea about the word walk and was simply observing the body's behavior, said without hesitation, "I am going to get a Coke." The interpreter did not say, "I do not know." It did not say, "Something is making me walk and I have no idea what." It generated a perfectly reasonable story, delivered it with complete confidence, and believed it entirely. Now, this was a split-brain patient, so the interpreter was working with unusually limited information. But Gazzaniga's argument is that the same process happens in all of us all the time. The vast majority of neural processing happens below the level of conscious awareness. Actions and decisions are generated by distributed networks operating in parallel. And the interpreter, sitting in the left hemisphere, watching the output, constructs a story about why you did what you did, who you are, and what you intend to do next. Most of the time, the story is close enough to reality that it works. But it is always, at some level, a reconstruction, not a direct observation, but an interpretation. This does not make the story false in any meaningful sense. A good interpretation, like a good map, is useful precisely because it simplifies. A map that was a perfect replica of the territory would be as big as the territory and therefore useless. The interpreter's narrative about who you are and why you do things is useful because it compresses an unimaginably complex neural reality into something you can work with, something you can communicate to other people, something you can plan around. But it is worth knowing, as you lie here drifting towards sleep, that the version of yourself you carry around in your head is a sketch, not a photograph. A working model, not the thing itself. The real you is bigger, deeper, more complex, and more mysterious than any story could capture. But the implications are profound.
Because if cutting the brain in half can reveal two separate minds that were always there, working in parallel, then what about all the other divisions? The brain has dozens of specialized regions, hundreds of distinct circuits, billions of neurons organized into networks that we are only beginning to map. How many of those networks are doing their own thing, running their own computations, making their own judgments, all without the conscious you having any idea? The answer, as it turns out, is most of them. Let's come back to your body for a moment. Right now, as you lie here, there is a war going on inside your autonomic nervous system, and you are completely unaware of it. Or rather, it is not exactly a war. It is more like a negotiation, a constant tug-of-pull between two forces that are adjusting the settings of your internal organs moment by moment. On one side is the sympathetic nervous system. This is the part that ramps things up. It increases your heart rate, dilates your pupils, redirects blood from your digestive system to your muscles, releases adrenaline. This is the fight or flight system, the one that prepares your body for action, for running, for fighting, for responding to threats. When you see something frightening, when you feel stressed or anxious, your sympathetic system kicks in. On the other side is the parasympathetic nervous system. This is the one that calms things down. It slows your heart rate, constricts your pupils, stimulates digestion, promotes salivation. This is the rest and digest system, the one that tells your body the coast is clear, that it is safe to relax and conserve energy. These two systems talk to the same organs but exert opposite effects. Your heart receives signals from both. Your intestines receive signals from both. Your salivary glands, your bronchial tubes, your bladder, all of them are being constantly tuned by these two opposing forces. And the balance between sympathetic and parasympathetic activity shifts all the time, depending on what you are doing, what you are feeling, what is happening around you. Here is what matters for our story. You have absolutely no voluntary control over this. You cannot decide to speed up your heart by thinking about it. You cannot tell your stomach to stop digesting. You cannot instruct your pupils to dilate. All of this is handled automatically by brain circuits that operate entirely below the level of consciousness. The brainstem and the hypothalamus are the main command centers, and the vagus nerve, the longest cranial nerve in your body, is the main information highway. The vagus nerve is worth knowing about. Its name comes from the Latin word for wandering, and it earned that name because early anatomists were astonished by how far it reaches.
It originates in the brainstem, exits the skull, and then wanders down through your chest and into your abdomen, sending branches to your heart, your lungs, your esophagus, your stomach, your intestines, and a host of other organs. It carries both motor signals, telling the organs what to do, and sensory signals, reporting back to the brain on the state of your internal world. When you feel that uncomfortable pressure after eating too much, that is your vagus nerve reporting that your stomach is distended. When you hiccup, that is a vagal reflex, triggered by excessive stretching of the stomach. When your heart slows down as you relax into sleep, that is the parasympathetic branch of the vagus nerve, taking over from the sympathetic system. And right now, as your body shifts towards sleep, the parasympathetic side is winning the negotiation. Your heart is slowing. Your breathing is deepening. Your digestive system is waking up, doing its quiet work. The sympathetic system is dialing down. All of this without a single conscious instruction from you. The stranger is at work. But the autonomic nervous system is just one example of brain activity that runs without your awareness. Consider something even more dramatic. Consider what happens to your brain when you sleepwalk. Sleepwalking is surprisingly common. Studies suggest that somewhere around 15% of children and about 4% of adults sleepwalk at least once. For most people, it is occasional and harmless, a brief episode of wandering or mumbling. But for some people, it is far more elaborate. There are documented cases of sleepwalkers cooking meals, painting walls, even driving cars. One woman reported waking up to find that she had painted a perfect triangle on her kitchen wall during the night. When she saw it in the morning, she laughed because she had no memory of doing it, and she could not figure out how she had managed it while unconscious. What is happening in the brain during sleepwalking is both simple and deeply strange. During sleep, your brain cycles through phases of high and low activity. In the deepest phase, called slow-wave sleep, your brain waves become large and slow, and most of your cerebral cortex is essentially offline. But in some people, during this deep sleep phase, there is a sudden partial arousal. Parts of the brain wake up while other parts stay asleep, and here is the critical detail. The parts that wake up are the motor cortex, which controls movement, the visual cortex, which processes visual information, and the coordination centers in the cerebellum and brainstem. The parts that stay asleep include the prefrontal cortex, the region responsible for deliberate decision-making, self-awareness, and the conscious experience of being you. So the sleepwalker can stand up, navigate around furniture, open doors, walk downstairs, and even perform complex tasks like painting. The motor system works. The visual system works. Balance works. But the decision maker, the part of the brain that says, "I am choosing to do this," is still in deep sleep. The person is acting without agency, without self-awareness, without conscious intention. And when they wake up, they have no memory of it, because the memory-forming systems were also not fully engaged. Sleepwalking reveals something important about the nature of consciousness. Consciousness is not an all-or-nothing switch. You are not simply conscious or unconscious. Different parts of your brain can be in different states at the same time. You can have a brain that is awake enough to walk, but not awake enough to know it is walking. The machinery of action can operate independently of the machinery of awareness, which means that a lot of what you do during your waking hours might also be less conscious than you think. Consider driving. You have probably had the experience of arriving at a destination and realizing you have no memory of the last several minutes of the drive. You were not asleep. Your eyes were open. You were steering. You were braking at stop signs. You were making decisions. But the you that monitors and narrates experience had checked out. The driving was handled by something else, some combination of motor memory and habit and automatic processing that did not require your conscious attention. Or consider walking. The act of walking is extraordinarily complex. Just teaching a robot to walk on two legs without falling is one of the hardest problems in engineering. It requires constant real-time adjustments to balance, weight distribution, muscle tension, joint angle, ground surface. Your brain handles all of this without any conscious input from you. You do not think about how to walk. You just walk. The computational work is done by circuits in your cerebellum, your basal nuclei, and your spinal cord, and none of it reaches your awareness. The list goes on. Regulating your heart rate, adjusting your blood pressure, releasing hormones, processing visual input to maintain balance, filtering sensory information to decide what gets your conscious attention and what gets ignored, generating your next breath, running your immune system, coordinating the muscles in your throat when you swallow. Every single one of these operations is handled by the stranger, by the vast, unconscious machinery that does its work silently and efficiently, all day and all night, without you ever knowing it. A neuroscientist named David Eagleman has put it this way: your consciousness is like a tiny stowaway on a transatlantic steamship who takes credit for the entire journey. The conscious you, the narrator, the one who feels like it is in charge, is really just a small part of a much larger operation. Most of the brain's work happens in the dark. But if so much of your brain's work is unconscious, then what exactly is consciousness for? And how does it relate to the deepest and most mysterious operation your brain performs, the one that we call memory? Before we talk about the hippocampus and memory, there is something worth understanding about how the brain processes sensory information, because this is where the question of who is in control becomes particularly vivid. Right now, your senses are active, even as you rest. Your ears are picking up the ambient sounds in the room. Your skin is registering the temperature and texture of the bedding. Your proprioceptive system, the network of sensors in your muscles, joints, and tendons, is continuously reporting the position of your body to your brain. Even with your eyes closed, you know exactly where your arms and legs are. You could reach out and touch your nose without any difficulty, because your parietal lobe is maintaining a constantly updated map of your body's configuration in space. Here is what is remarkable about all of this. Only a tiny fraction of the sensory information reaching your brain ever makes it to conscious awareness. Your retinas alone send about 10 million bits of information per second to the visual cortex. Your ears, your skin, your muscles, your internal organs, they are all generating their own streams of data. The total flow of sensory information into your brain is staggering, and yet you are consciously aware of only a minuscule sliver of it. The rest is handled by the stranger. Your brainstem, your thalamus, and various cortical areas are filtering, sorting, prioritizing, and discarding the vast majority of the incoming data before it ever reaches the level of conscious processing. The thalamus, in particular, acts as a gatekeeper.
Almost all sensory information, with the notable exception of smell, passes through the thalamus on its way to the cortex. And the thalamus does not just relay the signals passively. It actively selects which signals to amplify and which to suppress, based partly on what the cortex has told it to look for, and partly on the intrinsic properties of the signals themselves. New signals get priority. Strong signals get priority. Signals that match a predicted threat get priority. Everything else gets turned down. This is why you can be in a noisy restaurant and still follow a conversation. Your thalamus and your auditory cortex are collaborating to boost the signal of the voice you are attending to and suppress the background chatter. And this is also why a sudden loud noise, even in the middle of sleep, can jolt you awake. The thalamus never fully closes the gate on intense or novel stimuli. Your alarm system is always on, even when the rest of you has gone dark.
In the midbrain, there is a structure called the superior colliculus, part of the four little bumps known collectively as the tectum, or the quadrigeminal plate. The superior colliculus is one of the most ancient visual processing centers in the brain, and in fish and amphibians, it is the primary visual center, the place where visual information is processed and acted upon. In humans, most visual processing has migrated up to the occipital cortex, but the superior colliculus still plays a crucial role. It detects new visual stimuli, especially moving objects in the periphery, and it triggers a rapid orienting response, turning the eyes and head toward the stimulus before you have consciously registered what it is. Ivan Pavlov, the Russian physiologist best known for his work on conditioning, called this the orienting reflex, and he considered it one of the most fundamental behaviors in the animal kingdom. When something new appears in the environment,
You turn to look at it. This is not a learned behavior. It is built into the brain stem. It is curiosity at its most primitive level. The brain's innate drive to gather information about changes in the environment. And it operates entirely below the level of conscious control. By the time you become aware of the new stimulus, your eyes have already moved toward it. So the processing of sensory information is, from start to finish, largely an unconscious affair. The stranger does the filtering, the prioritizing, the rapid responding. Consciousness gets the edited highlights, a polished summary of what the unconscious systems have already decided is important. This raises an interesting question about perception itself. What you perceive is not the raw world. What you perceive is the world as your brain has constructed it after extensive editing and interpretation. Colors, for example, do not exist in the physical world. What exists is electromagnetic radiation of different wavelengths. Your visual system converts those wavelengths into the experience of color, and it does so in ways that are surprisingly influenced by context. The same wavelength of light can appear as different colors depending on the surrounding colors, the lighting conditions, and your brain's assumptions about what the object should look like. The solid world around you, everything that feels firm and real and substantial, is also a construction. At the atomic level, matter is overwhelmingly empty space. The feeling of solidity comes from electromagnetic forces between atoms, translated by your sensory systems into the experience of touching something hard or soft, rough or smooth. Your brain takes the raw sensory data and builds a model, a simulation, that is useful for navigating the world, but that is not a literal copy of physical reality. So even your most basic experience of the world, the experience of seeing and hearing and touching, is mediated by the stranger.
The conscious you lives in a world that has been pre-processed, filtered, edited, and constructed by unconscious systems. The stranger builds the stage. Consciousness walks out and performs on it. There is a sense you use every waking moment that most people never think about, and it is one of the most beautiful examples of the stranger's handiwork. It is called proprioception, and it is the sense that tells you where your body is in space. Close your eyes. Now touch your nose with your fingertip. You did it easily, without thinking. But consider what that simple act required. Your brain needed to know the current position of your arm, the angle of your elbow, the extension of your finger, and the location of your nose relative to the rest of your body. None of this information came from vision, since your eyes were closed. It came from thousands of tiny sensors embedded in your muscles, tendons, joint capsules, and ligaments. Sensors that are continuously reporting on the degree of stretch in every muscle, the angle of every joint, the tension in every tendon. Your cerebellum collects all of this information, integrates it, and maintains a constantly updated three-dimensional model of your body's position. And here is the thing that makes proprioception so relevant to our story. You are almost never conscious of it. Right now, your brain knows exactly where your legs are, how your arms are positioned, whether your head is tilted, how your spine is curved. It knows all of this with remarkable precision. But unless you direct your attention to it, none of this knowledge reaches your conscious awareness. The information flows continuously from the sensors in your body to the processing centers in your cerebellum and parietal lobe, and the model is updated moment by moment, and you have no idea any of it is happening. When I mentioned it just now, when I asked you to think about the position of your legs, you suddenly became aware of something that was always there, just below the surface. The information was available to consciousness all along, but it was being processed at a level that did not require your attention. The stranger was handling it. This is actually a general principle of how the brain manages attention. There is vastly more information available at any given moment than consciousness can handle. So the brain has evolved a system of prioritization. The thalamus, the prefrontal cortex, and the anterior cingulate cortex work together to select which streams of information will be boosted into conscious awareness and which will remain in the background. Most sensory information is processed and acted upon without ever becoming conscious. You feel the floor under your feet without thinking about it. You adjust your posture without deciding to. You blink, swallow, and breathe without awareness. All of this is the stranger, managing the routine so that the conscious mind is free to focus on whatever matters most in the current moment. And as sleep approaches, the range of information that consciousness attends to shrinks further and further. The thalamus gates close, the prefrontal cortex powers down. Proprioceptive signals continue to flow, but they no longer produce the sharp awareness they would during the day. Your body is still being monitored, still being maintained, but the conscious mind is withdrawing, leaving the stranger to run the show alone. Which is, when you think about it, an extraordinary act of trust.
Every night, you surrender conscious control of your body to a set of brain systems that you have never met and will never meet. And you do it willingly, automatically, without a second thought. You trust the stranger with your life, and the stranger has never once let you down. Let us stay here for a while, because memory is where things get really personal. Your memories are, in a very real sense, who you are. The continuity of your identity, the feeling that you are the same person who woke up this morning, the same person who went to school as a child, the same person who has a specific set of likes and dislikes and fears and attachments. All of that is held together by memory. Without memory, there is no narrative. Without narrative, there is no self. And the most dramatic demonstration of this comes from a man named Henry Molaison, who spent most of his life known to the scientific world only by his initials, H.M. Henry suffered from severe epilepsy. His seizures were frequent and debilitating, and by the time he was in his late 20s, in 1953, the standard medications were not working. His doctors decided to try surgery. The region of the brain that seemed to be generating his seizures was the medial temporal lobe, and specifically the hippocampus. So a surgeon removed Henry's hippocampus, along with some surrounding tissue, from both sides of his brain. The surgery worked. His seizures improved dramatically. But the consequences were devastating. Henry lost the ability to form new long-term memories. After the surgery, Henry could remember everything from his life before the operation. He could talk about his childhood, his family, events from his earlier years. His personality was intact. His intelligence was intact. He could hold a conversation, follow a joke, understand what was happening around him. But the moment something left his immediate attention, it vanished. If you introduced yourself to Henry and then left the room for 5 minutes and then came back, he would have no idea who you were. You would need to introduce yourself again, and again the next time, and the next. The doctors and researchers who studied Henry for decades faced this reality every time they walked into his room. They had known him for years. He had no idea who they were. Each visit was, for him, the first. But here is where things become truly fascinating, because Henry's memory loss was not total. It was selective, and the pattern of what was preserved and what was lost tells us something profound about how memory is organized in the brain. Henry could not remember meeting you. He could not remember what he had for breakfast. He could not remember a conversation from 10 minutes ago. But he could learn new motor skills. If you taught him to trace a shape while looking at it in a mirror, which is a tricky task that requires practice, Henry would improve over multiple sessions. Each time he sat down to do the task, he would say he had never done it before. He had no conscious memory of the previous sessions, but his hands knew. His performance improved, just as it would for anyone else. Somewhere in his brain, a record of the practice was being laid down, even though it never reached his conscious awareness. And there was another remarkable detail. Henry lived in the same house for years after his surgery, the same layout, the same rooms, the same doorways. And if you asked him to draw a floor plan of that house, he could do it perfectly. He knew where every room was, how they connected, where the doors and windows were.
He could not tell you when he had learned this information, could not point to a specific memory of walking through the house, but the knowledge was there, embedded in some other system. This tells us that memory is not one thing. There are multiple memory systems in the brain, and they are at least partially independent of each other. The hippocampus, which was removed from Henry's brain, is critical for what we call declarative memory, the conscious, explicit memory of facts and events. This is the memory system that lets you recall what you had for dinner, or where you parked your car, or the name of the person you just met. Without the hippocampus, new declarative memories cannot be formed. But procedural memory, the memory of how to do things, is handled by different circuits, primarily in the cerebellum and the basal nuclei. These circuits were intact in Henry's brain, which is why he could learn new motor skills without being aware of it. And spatial memory over very long periods, the kind of deep familiarity with a place that comes from years of living in it, seems to be stored in yet another way, distributed across cortical networks that do not depend on the hippocampus for retrieval once the memories are firmly established. Now, how does the hippocampus normally do its job? How does it take a fleeting experience and turn it into a lasting memory? The mechanism involves those NMDA receptors and glutamate synapses that neuroscientists have spent decades studying. In the hippocampus, there are synapses that have a special property. They can be strengthened by experience. Under normal conditions, an NMDA receptor on the postsynaptic neuron is blocked by a magnesium ion that sits in the channel like a plug. When a weak signal comes through, the plug stays in place and the signal is not particularly well transmitted. But when a strong signal arrives, or when two signals arrive at the same time from different sources, the plug is knocked loose, the NMDA channel opens, calcium floods into the cell, and a cascade of molecular events is triggered that makes the synapse stronger. More receptors are inserted into the membrane, more neurotransmitter is released. The connection between those two neurons becomes more efficient. This process is called long-term potentiation, and it is one of the main cellular mechanisms of learning and memory. When you remember something, what has physically happened in your brain is that a specific pattern of synapses has been strengthened, so that when part of the pattern is activated, the rest of the pattern lights up along with it. A memory is a pattern of synaptic strengths, and the hippocampus is the structure that orchestrates the initial formation of that pattern. But the hippocampus is not where long-term memories ultimately live. Its job is more like that of a temporary staging area. During the day, the hippocampus encodes new experiences, tagging them, holding them, keeping them fresh. And then, during sleep, something remarkable happens. The hippocampus replays the day's experiences and gradually transfers them to the cerebral cortex for long-term storage. The magnesium plugs return to the NMDA receptors, the hippocampus resets, and the memories that have been transferred to the cortex become part of the permanent record. This is why sleep matters so much for memory. During slow-wave sleep, the hippocampus and the cortex engage in a kind of dialogue, replaying and consolidating the day's experiences. If you do not sleep, or if your sleep is disrupted, this consolidation process is impaired, and memories that should have been transferred are lost. Every night, while you are unconscious, your hippocampus is editing your autobiography, deciding what to keep and what to discard.
And you have no say in the matter. Here is a detail that I find particularly striking. Researchers have shown that you can disrupt hippocampal memory by applying a brief electrical stimulus, like a mild shock, that causes all the NMDA channels to open at once. When this happens, the hippocampus is essentially wiped, and the person loses not only the memory of the shock itself, but also memories from several hours before the shock. This is called retrograde amnesia, and it tells us that the memories stored in the hippocampus during the day are fragile, held in place by those strengthened synapses, and vulnerable to disruption until they have been consolidated during sleep. So memory, like consciousness, is not one thing. It is multiple systems, partially overlapping, partially independent, each handling a different aspect of what it means to remember.
And the feeling you have of a single continuous memory, a single unbroken thread of personal history, is another construction, another collaboration between systems that are, underneath, doing very different things. Henry Molaison died in 2008, at the age of 82. By that time, he had been studied by more neuroscientists than almost anyone in history. He never remembered any of them. He lived in a permanent present, unable to carry forward anything that had happened since 1953, unable to update his sense of who he was or what his life had become. His sense of self was frozen in time, a young man's identity persisting in an aging body. And that frozen identity, more than anything else, shows us what memory does. It does not just record the past. It makes the present intelligible. It makes you, you.
There is a deeper story here about how memory works at the cellular level, and it is worth taking the time to walk through it, because it connects back to some of the fundamental questions we have been exploring tonight about the nature of identity and the boundary between the conscious and the unconscious. We talked about long-term potentiation, the process by which synapses are strengthened through repeated use. But LTP is really just the middle tier of a three-level system. At the most basic level, there is a process that a Nobel Prize-winning neuroscientist named Eric Kandel studied in an organism that could not be less glamorous, a large sea slug called Aplysia. Kandel chose Aplysia because its nervous system is, by brain standards, absurdly simple. It has about 20,000 neurons compared to our 86 billion, and many of those neurons are large enough to see with the naked eye and to insert electrodes into individually.
Kandel could map the exact circuit responsible for a specific behavior, a defensive reflex in which the animal pulls its gill inside a protective pouch when the gill is touched, and then watch what happened at each synapse in the circuit when the animal learned something. What Kandel discovered was a mechanism he called sensitization, or what we might more broadly call summation. If you give Aplysia a very gentle touch on the gill, too light to trigger the withdrawal reflex, nothing happens. But if you repeat that gentle touch every few seconds, eventually the animal does respond. The reflex fires and the gill is pulled in. The mechanism is elegant and simple. Each time the touch signal passes through the relevant synapse, a small amount of calcium flows into the axon terminal. Calcium is needed for the release of neurotransmitter, and under normal circumstances, it is pumped back out after the signal has passed. But if the signals come quickly enough, faster than the calcium can be removed, the calcium accumulates. With each successive touch, there is a little more calcium in the terminal, a little more neurotransmitter is released, and the signal transmitted across the synapse gets a little stronger. Eventually, it is strong enough to trigger the reflex. This is the most basic form of memory. It lasts only as long as the calcium remains elevated, typically a matter of minutes. Once the stimulation stops and the calcium is pumped out, the synapse returns to its baseline state and the memory is gone. This is why you can repeat a phone number to yourself and remember it long enough to dial, but if someone interrupts you, it evaporates. The calcium disperses and the temporary trace vanishes. The second level is long-term potentiation, which we discussed earlier. LTP involves the NMDA receptors and can last for hours.
The magnesium plug is knocked out of the channel. Calcium flows in through a different route, and the synapse is temporarily strengthened in a more robust way. This is the mechanism that the hippocampus uses to hold on to the day's experiences until they can be consolidated during sleep. But neither of these mechanisms can explain truly permanent memories, the kind that lasts for years or decades. For that, you need the third level, which involves changes to gene expression. And this is where the story gets deeply interesting. Konrad Lorenz, the Austrian zoologist who won the Nobel Prize in the 1970s, described a phenomenon he called imprinting. When a baby chick or gosling hatches, it goes through a brief window of time, usually the first day or two of life, during which it attaches to whatever moving object it sees. Normally, that object is its mother. But Lawrence famously showed that if you remove the mother and substitute yourself, the chick will imprint on you instead.
It will follow you around, treat you as its parent, and direct all of its baby behaviors toward you. The neural mechanism of imprinting turns out to be genuinely remarkable. In the relevant brain region, there are synapses that are structurally ready to transmit signals, but that have very few receptors on the postsynaptic side. They are like doors that are built but not yet fully opened. When the chick sees the object it is going to imprint on, the visual signal passes through these synapses over and over, and each time a small signal reaches the nucleus of the postsynaptic neuron. Eventually, enough signal accumulates to activate specific genes that code for glutamate receptors. The cell begins manufacturing new receptor proteins. These proteins are transported to the synapse and inserted into the membrane. Now the synapse has more receptors, it transmits more efficiently, and the connection is permanently strengthened. This is a fundamentally different kind of memory from the first two. Summation changes how much calcium is present in the terminal. LTP changes whether a channel is open or blocked. But imprinting changes the physical structure of the synapse by adding new proteins. And because those proteins are being manufactured from the cell's own DNA, the change can be maintained indefinitely. This is how permanent memories are formed. The experience reaches all the way down to the genome, activates specific genes, produces new proteins, and physically rebuilds the synapse. The same basic mechanism underlies the formation of all long-term memories, not just imprinting. When you learn a new word, when you remember a face, when you master a skill, the process involves, at the deepest level, changes in gene expression that alter the structure of synapses. Your experiences literally reshape the physical matter of your brain. And this takes us back to the question of identity. If your memories are patterns of synaptic strengths, and if those strengths are determined by experience-driven changes in gene expression, then who you are is, at the molecular level, a record of everything that has happened to you. Every important conversation, every emotional event, every repeated practice session, every significant relationship, all of it has left a physical trace in the structure of your neurons. You are, quite literally, made of your experiences. The philosopher John Locke argued in the 17th century that personal identity is constituted by memory, that you are the same person as your earlier self because you remember being that person. Modern neuroscience supports this in a surprisingly concrete way. The continuity of your identity is maintained by the physical continuity of your synaptic patterns. If those patterns were erased, as they were in Henry Molaison's hippocampus, the thread of identity is broken. The past self becomes unreachable, and the present self cannot lay down new connections to the future. If you are still here, just let that settle. And if you have been drifting in and out, that is fine too. Everything we have talked about, the neurons, the synapses, the hemispheres, the hippocampus, all of it is working inside you right now, doing exactly what it was designed to do. We are not in a hurry. Because now we are going to turn to something that brings all of these threads together. We are going to talk about emotions and decisions and the deeply surprising relationship between the two. For most of the history of Western thought, emotion and reason have been treated as opposites. Emotion was the unruly force that pulled you away from good decisions, and reason was the noble faculty that kept you on track. But as neuroscientists began to study the brain circuits involved in decision-making, they discovered something that turned this entire picture upside down. Let's go back to Phineas Gage for a moment. After the accident, Gage did not just become rude and impulsive, he also became unable to make good decisions. He would make plans and then abandon them. He would start projects and lose interest. He could analyze a situation, weigh the options, understand the consequences, and still make the wrong choice. His reasoning skills in isolation seemed functional, but when it came to actually choosing what to do in real life, something was broken. For more than 100 years, no one could explain this. And then, in the 1990s, a neuroscientist named Antonio Damasio studied patients with damage similar to Gage's. Damage to the ventromedial prefrontal cortex, that region just behind the forehead. Damasio found that these patients shared a striking pattern. They performed normally on standard tests of intelligence and reasoning. They could solve logic puzzles, answer factual questions, score well on IQ tests. But in their actual lives, they made terrible decisions. They entered into bad business deals. They alienated their friends and families. They could not hold down jobs or maintain relationships. They seemed to understand, intellectually, what the right choice was, but they could not feel it. Damasio proposed something that was controversial at the time, but has since been supported by a great deal of evidence. He argued that emotions are not the opposite of reason. Emotions are essential to reason. Specifically, he suggested that when you are faced with a decision, your brain does not just analyze the options logically. It also generates a set of bodily signals, gut feelings, emotional responses that are based on your past experience with similar situations. These signals, which Damasio called somatic markers, bias your decision-making in the right direction, often before you are even consciously aware of the analysis. When something feels wrong, that feeling is your brain pulling up the emotional record of past experiences and using it to guide you. Patients with damage to the ventromedial prefrontal cortex have lost access to these somatic markers. They can still reason, but they cannot feel their way to a decision. And it turns out that feeling your way to a decision is, in many situations, more important than thinking your way to one. Because the world is too complex, too ambiguous, and too fast-moving for pure logical analysis to work in real time. Emotions are not noise in the system. They are a computational shortcut, a way for your brain to compress millions of past experiences into a quick signal that says, do this, or don't do that. A neuroscientist named Luke Chang has studied this at the level of brain circuits, using a game from behavioral economics called the trust game. In this game, one person has a sum of money and can choose to invest some of it with a partner. The investment is multiplied, and then the partner decides how much to return. The partner could keep all the money, which would be the purely selfish choice, or they could return a fair share, which requires overcoming the temptation of greed. Chang scanned people's brains while they played this game, and he found that the decision to be generous, to return a fair share, was driven in large part by a brain region called the insula. The insula is a patch of cortex tucked deep inside the lateral sulcus, the groove that separates the frontal and temporal lobes. Chang describes it as a kind of internal thermometer. The insula monitors your bodily states, your gut feelings, your sense of discomfort, and it generates signals that say, "This does not feel right," or "I would feel terrible if I did that." When people in the trust game considered keeping all the money, the insula lit up with activity, generating a signal that Chang interprets as guilt, or the anticipation of guilt.
And this signal pushed them toward cooperation. It made them return a fair share, not because they calculated that cooperation was strategically optimal, but because the thought of being unfair made them feel bad in their bodies. But the insula is just the thermometer. The information it generates needs to go somewhere to be acted on, and that somewhere is the prefrontal cortex, specifically the medial and ventromedial prefrontal cortex, which Chang describes as the thermostat. The thermostat takes the temperature reading from the insula and uses it to regulate behavior, to turn the emotional signal into an actual decision. And now you can see what happened to Phineas Gage. The iron rod destroyed his thermostat. The insula was probably still generating signals, still producing gut feelings about what was appropriate and inappropriate, what was fair and unfair, what was risky and safe. But the thermostat, the prefrontal region that translates those feelings into behavior, was gone.
Gage could feel at some level, but he could not use those feelings to guide his actions. The emotional rudder had been ripped away, and he was adrift. There is a powerful illustration of this emotional guidance system that comes from everyday experience, and it is worth sitting with because it connects to something very old in the brain's architecture. Have you ever met someone for the first time and had an immediate sense that something was off? Not based on anything they said or did, but just a feeling in your body, a vague discomfort, a reluctance to trust. That feeling is your brain pulling up a vast database of past social interactions, matching the new person's facial expressions, tone of voice, body language, and other subtle cues against patterns stored in your memory, and generating an emotional summary that arrives in your consciousness not as a reasoned judgment, but as a bodily sensation. The feeling is the output of a computation that you cannot see and did not consciously perform. This is the somatic marker at work. And the reason it is so fast and so effective is that the brain regions involved, the amygdala, the insula, the anterior cingulate cortex, the ventromedial prefrontal cortex, are all connected in tight loops that can process social and emotional information in milliseconds. Long before your slow, deliberate verbal reasoning system has finished analyzing the situation, the fast, automatic, emotional system has already delivered its verdict. The anterior cingulate cortex, that strip of tissue running along the inner surface of the hemispheres just above the corpus callosum, deserves special mention. This region acts as a kind of evaluator for ongoing behavior. When you are pursuing a goal and something goes wrong, the anterior cingulate detects the error and generates a signal that feels like frustration or disappointment, or the nagging sense that you need to change your approach. When something goes right, it generates a signal that feels like satisfaction or relief, or the sense that you are on the right track. A Russian physiologist named Pyotr Simonov proposed that emotions in general can be understood as the brain's way of evaluating progress toward goals. Positive emotions arise when progress is better than expected. Negative emotions arise when progress is worse than expected. And the intensity of the emotion corresponds to the importance of the goal. This framework, while simplified, captures something essential about the function of emotions in decision-making. They are not random noise. They are not primitive impulses that need to be suppressed by reason. They are the brain's real-time assessment of how well your life is going, computed by circuits that have been refined by hundreds of millions of years of evolution. And these circuits never sleep. Even as you lie here, drifting toward unconsciousness, your emotional brain is still active, still monitoring, still ready to wake you with a jolt of anxiety if something threatens you, or to suffuse your dreams with the unresolved feelings of the day. The stranger keeps watch even when the conscious mind has gone dark. Think about your own experience for a moment. When you are deciding whether to say something that might hurt someone's feelings, you do not typically perform a logical analysis of the social consequences. You feel something. A tension in your chest, a tightening in your gut, a sense of hesitation. That feeling is your insula and your prefrontal cortex working together, pulling up emotional data from past experiences and translating it into a behavioral recommendation.
And most of the time, you follow the feeling, without ever being consciously aware of the computation that produced it.
This is the stranger at work again. The emotional guidance system operates largely outside of consciousness. You experience its output, the feeling of unease, the sense that something is wrong, but you do not experience the process. The processing happens in the basement. You just get the results delivered to the main floor. And emotions shape your decisions in ways that go far beyond simple gut feelings. Other people's emotions shape your decisions too. Your brain has evolved to be exquisitely sensitive to the emotional states of the people around you. A large fraction of your cortex is devoted to reading facial expressions, interpreting body language, inferring what other people are thinking and feeling. This capacity, sometimes called theory of mind, is one of the things that makes human social life possible. You do not just react to what people do,
You react to what you think they feel.
Neuroscientist Heather Berlin has pointed out that how you make decisions, how you behave, how you think about yourself, all of these processes are developed by interacting and synchronizing with other brains. Your brain does not operate in isolation. Every experience you have, every sentence you hear, every image you see, changes the physical structure of your brain, however slightly. The people around you, their expectations, their emotions, their behavior, all of this is constantly molding your neural circuits. We are social animals at the deepest neurological level. The stranger in your head is not just a solo operator. It is a social creature, constantly adjusting itself to the social world. But if the stranger is doing so much of the work, steering your emotions, guiding your decisions, monitoring the social landscape, then what is left for the conscious you to do? And how reliable is that feeling you have of being in control?
Let's sit with that question for a moment.
Breathe. Let the room be quiet. Because this next part is where things get really strange. Let's slow down here for a moment and breathe. We have been traveling through some deep territory, and there is no need to rush. If you have been drifting in and out, just let whatever you caught settle naturally. The brain does not need you to consciously organize this information. It will do that work on its own, in the background, the way it always does. Right now, your brain is in a state of transition. The waking networks are starting to hand off to the sleeping networks. The neuromodulators that keep you alert, the norepinephrine from the locus ceruleus, the serotonin from the raphe nuclei, the acetylcholine from the basal forebrain, all of these are beginning to decrease their output. And the sleep-promoting signals from the ventrolateral preoptic area of the hypothalamus are ramping up, inhibiting the arousal centers, allowing the slow waves of sleep to spread across the cortex. Your brain knows how to do this. It has done it thousands of times. You do not need to help it. Just let it happen. When we talked about the split-brain patients, about Phineas Gage, about the autonomic nervous system, about memory and the hippocampus, we were building towards something. We were building toward a picture of the brain as a collection of semi-independent systems, each doing its own work, each contributing to the larger whole, but none of them constituting the real you by itself. The conscious you, the narrator, the interpreter, is just one system among many, and it is not even the most important one. This might sound like a diminishment.
It might sound like we are saying that consciousness does not matter, or that free will is an illusion, or that you are just a machine running on autopilot. But that is not what the science says. What the science says is something subtler and, I think, more beautiful. Consciousness matters. The interpreter matters. The narrator matters. Without the capacity for self-reflection, for deliberate planning, for moral reasoning, you would not be able to live a human life. The prefrontal cortex, the interpreter, the executive functions, these are what allow you to set long-term goals, to resist temptation, to learn from mistakes, to imagine the future, to care about other people's feelings. These are not trivial abilities. They are what make human civilization possible. But the conscious mind is most effective when it works in partnership with the unconscious systems, rather than trying to override them. The best decision makers are people who combine analytical thinking with emotional intuition. The best athletes are people who practice consciously until a skill becomes automatic and then trust the automatic system to perform. The best creative artists are people who master their craft through years of deliberate practice and then learn to let go, to let the unconscious systems generate new combinations and connections that the conscious mind alone could never produce. The relationship between the conscious and unconscious parts of you is not a battle. It is a collaboration, and the strange, beautiful, slightly unsettling truth about being human is that you will never fully know the collaborator. You will never meet the stranger face to face, but you can learn to trust it, because it has been on your side from the very beginning. Now, let us talk about what happens when scientists tried to measure the exact moment when a decision is made. Because this is where the question of control becomes as sharp and as personal as it will ever get. A neuroscientist named Uri Maoz has spent years studying the sense of agency, that feeling you have that you are the one initiating your own actions. And to show how fragile that feeling can be, he starts his experiments with a magic trick. When Heather Berlin, a clinical psychologist and neuroscientist, visited Maoz's lab for a documentary, he handed her a sealed envelope at the very beginning of their meeting. He told her not to open it, just hold on to it. Later, he gave her a book and asked her to flip to any page she wanted and find a word that had some meaning for her. She chose the word representation on page 105. She wrote it down on a sticky note. At the end of the session, Maoz asked her to open the envelope. Inside was a card. On the card was written, "You will choose the word representation on page 105. It was, of course, a magic trick. Magicians use misdirection, forced choices, and other techniques to make people think they are choosing freely when they are actually being guided. But the point Maoz was making was not about magic. The point was about the fragility of the feeling of choice. Berlin felt with complete certainty that she had freely chosen that word. She was shocked when the envelope revealed that her choice had been predicted or engineered without her awareness. Maoz's real experiment goes deeper. He uses a device called transcranial magnetic stimulation, or TMS, which generates a focused magnetic field that can stimulate specific parts of the brain through the skull. If you aim the TMS device at the part of the motor cortex that controls your fingers, you can make a person's finger twitch involuntarily. The person feels the finger move, but they did not initiate the movement. Their brain sent the signal because it was externally stimulated, not because of any conscious decision. In the experiment, a volunteer named Tomasz sat with the TMS device positioned over his motor cortex. Sometimes the researchers asked him to move his finger on his own.
Sometimes the researchers used the TMS to move his finger for him. And sometimes, at the exact moment Tomasz decided to move his finger voluntarily, the researchers also triggered the TMS, so that the finger moved from a combination of his own decision and the external stimulus. After each movement, Tomasz was asked 2 questions. Who initiated the movement, and how much agency did he feel over it? When Tomasz moved his finger on his own, without any TMS, he reported full agency. That was me. When the TMS moved his finger without his involvement, he said he had no agency. That was the machine. But in the critical condition, when Tomasz decided to move his finger, and the TMS also triggered the movement at the same instant, something remarkable happened. Tomasz did not say, "That was both of us."
He said, "I do not know," or "That was the machine." Even though he had genuinely decided to move his finger, even though the decision was entirely his, the addition of the external stimulus disrupted his sense of agency. He felt like he had not been in control, even though he was. Think about what that implies. The sense of agency, the feeling that you are the author of your own actions, is not a direct readout of what is actually happening in your brain. It is a perception, a construction, built from multiple sources of information, including motor commands, sensory feedback, timing, and expectations. And like any perception, it can be wrong. It can be disrupted. It can be manipulated. Maos puts it in terms that are worth sitting with. We walk around, and we feel like we are the authors of our actions, and you can see with just a little bit of messing around, that feeling tends to fall apart.
Our sense of self, our memories, our sense of agency, they are all things that our brain evolved over time. But they are fragile, and they can be manipulated under the right circumstances. This does not mean that you never have agency. In everyday life, your sense of being in control is generally accurate enough to be useful. You decide to pick up a glass of water, and your hand moves toward the glass, and the prediction matches the outcome, and so the feeling of agency is confirmed. The system works because, most of the time, the prediction and the outcome align. But the system is not infallible, and the fact that it can be disrupted tells us something important about its nature. Agency is not a metaphysical property of the soul. It is a neural computation, produced by specific circuits in the brain, and like all neural computations, it is subject to errors, biases, and illusions. There is an even more unsettling line of research that bears on this question. In the 1980s, a physiologist named Benjamin Libet conducted an experiment that has been debated ever since. Libet asked people to make a simple voluntary movement, a flick of the wrist, whenever they felt like it. While they did this, he recorded their brain activity using electrodes on the scalp. He also asked them to note the precise moment when they first felt the urge to move by watching a special clock. What Libet found was that the brain began preparing for the movement about half a second before the person reported feeling the conscious urge to move. There was a measurable buildup of electrical activity in the motor cortex, called the readiness potential, that preceded the conscious decision by several hundred milliseconds. The brain seemed to be making the decision before you knew about it. This experiment has been criticized on many grounds, and its interpretation is still debated. But the basic finding has been replicated many times, with more sophisticated methods. The conscious experience of deciding to act does seem to come after the neural processes that generate the action have already begun, which suggests that the conscious decision might be less of a cause and more of an after-the-fact report. The brain does something, and then consciousness catches up and takes credit. Maos has reflected on this too. He says that our experience of voluntary action is, in some sense, retrospective. The brain looks at what the body did, checks whether it matches the kind of thing the person would have chosen to do, and if it matches, stamps it with the label, "I did that." The sense of agency is a post hoc evaluation, not a real-time command. This is the interpreter again, that left hemisphere storyteller that Gazzaniga identified in the split brain patients. The interpreter does not just explain what the right hemisphere is doing, it explains everything.
It takes the stream of actions and experiences that pour through your life and weaves them into a coherent narrative with you as the protagonist. And most of the time, the narrative is close enough to reality that it works. But sometimes, if you look carefully enough, or if you disrupt the process with a TMS device, you can see the seams. Let's just pause here and breathe for a moment. None of this means that your life is an illusion or that you are not real. The experience of being you is as real as anything in the universe. But the mechanism that produces that experience is more complex, more distributed, and more surprising than it feels from the inside. You are not a single thing running the show. You are an orchestra. And the music emerges from the interplay of many instruments, most of which are playing without a conductor. And here is something that might seem counterintuitive, but is actually rather beautiful.
Sometimes the best thing the conscious mind can do is get out of the way. Charles Limb is a neuroscientist and surgeon who has spent years studying what happens in the brain during creative improvisation. In one of his experiments, he put musicians into an MRI scanner and asked them to do two things. First, play a piece of music from memory, a rehearsed, familiar piece. Then, improvise. Make something up on the spot, something they had never played before. The brain scans showed something fascinating. When the musicians were playing from memory, the prefrontal cortex, that decision maker, that self-monitor, was active. It was engaged, overseeing the performance, keeping things on track. But when the musicians began to improvise, something changed. The prefrontal cortex became less active, it quieted down, it stepped back. And this was especially pronounced in the most skilled improvisers.
The better someone was at making music up on the spot, the more their prefrontal cortex disengaged during improvisation. The decision maker was not just standing aside, it was actively being suppressed. In a particularly memorable version of this experiment, Lim scanned the brain of a freestyle rapper named Chris Emdin while he improvised lyrics in the scanner. Emdin had no idea what prompts he would be given. He would see a word on a screen and immediately begin rapping about it, making up rhymes and rhythms in real time. And the scans showed that same pattern. The prefrontal cortex went quiet, and other regions associated with motor planning, language production, and emotional expression, became more active. Lim interprets this as evidence that conscious self-monitoring can actually interfere with creative performance. When you are improvising, you need to let go of the part of your brain that evaluates and judges.
The inner critic, the part that says, "Is this good enough?" Or, "Am I going to make a mistake?" has to be silenced. Otherwise, it slows you down, introduces hesitation, and blocks the flow of ideas. And this is not just about music or rap. Every human being is creative in some way. All day long you are doing unscripted things. Conversations are improvised. Responses to unexpected situations are improvised. Life is not scripted, and so every person, in every moment, is balancing the need for conscious control with the need to let the unconscious systems do their work. Athletes know this well. A basketball player who has practiced free throws 10,000 times can make them almost automatically. But in a high-pressure situation, when the stakes are high and the player starts thinking about the mechanics of the throw, performance often drops.
This is called choking, and it happens because conscious attention to a well-practiced motor skill interferes with the automatic processing that would otherwise handle the skill beautifully. The prefrontal cortex, trying to help, actually makes things worse. So the stranger in your head, the unconscious machinery that handles most of your brain's work, is not just a background system that keeps the lights on. It is also the source of some of your best performances. Creativity, flow, athletic excellence, smooth social interaction. These all depend on the conscious mind learning when to lead and when to follow. And one of the hallmarks of expertise in any domain is the ability to hand control over to the unconscious systems that have been trained through years of practice. This might seem paradoxical. The whole story we have been telling tonight has been about the stranger, about the vast, unconscious systems that run without your awareness, and about how much of what you call you is really a construction, a narrative imposed on top of a much more complex reality. And you might think that the message is pessimistic, that you are not really in charge, that the feeling of being a unified person is just an illusion. But that is not quite right. The feeling of being you is real. It is produced by your brain, and your brain is real, and the experience it generates is real. The fact that the experience is constructed does not make it false. Your visual experience is also constructed. Your brain builds a three-dimensional model of the world from two-dimensional images on your retinas, filling in gaps, correcting errors, smoothing out the picture. And nobody says that vision is an illusion just because it involves construction. The same is true of the self.
You are a construction, but you are a magnificently useful one. The sense of unity, the feeling of being a single person with a continuous story, allows you to plan, to commit to long-term goals, to maintain relationships, to hold yourself accountable, to create meaning in your life. These are real functions, and they require the kind of coherent narrative that the interpreter provides. The insight from neuroscience is not that you do not exist. The insight is that you are more than you think you are. You are not just the voice in your head, the narrator, the conscious decision maker. You are also the heartbeat, the breath, the gut feeling, the motor skill, the spatial map, the emotional response, the memory that surfaces unbidden. You are the whole orchestra, not just the first violin. And sometimes the most productive thing the first violin can do is stop playing and let the rest of the orchestra be heard. This capacity for the unconscious to outperform the conscious is not limited to artistic improvisation or athletic performance. It shows up in domains you might not expect. Consider the experience of insight. You have probably had the experience of struggling with a problem, working at it consciously, getting nowhere, and then setting it aside. You go for a walk, or take a shower, or fall asleep. And then sometime later the answer appears, seemingly out of nowhere, fully formed. This is the aha moment, and it has been reported by scientists, mathematicians, writers, and composers throughout history. What seems to be happening is that when you stop consciously working on a problem, the unconscious processing does not stop. The relevant neural networks continue to explore combinations, to test associations, to sift through possibilities. But they do this in a way that is unconstrained by the assumptions and biases that conscious reasoning often brings. The conscious mind tends to get stuck in ruts, repeating the same approaches, following the same logical paths. The unconscious mind is free to wander, to make connections between ideas that are far apart, to try combinations that the conscious mind would reject as too unlikely. And sometimes, one of those unlikely combinations turns out to be the solution. When that happens, the result is pushed up into conscious awareness, and you experience it as a sudden insight, a flash of understanding that seems to come from nowhere. But it did not come from nowhere. It came from the stranger, working patiently in the dark while you were not paying attention. The German chemist August Kekulé famously reported that the structure of the benzene ring came to him in a dream, when he saw a snake biting its own tail. The French mathematician Henri Poincaré described solutions arriving suddenly while he was stepping onto a bus, after days of unsuccessful conscious effort. Otto Loewi, who discovered that nerve signals are transmitted chemically through neurotransmitters, literally dreamed the design of the experiment that would prove it. He woke up, scribbled it down, went back to sleep, and in the morning could not read his own handwriting. Fortunately, the dream came again the following night, and this time he got up and did the experiment immediately. These stories are not evidence that the unconscious mind is mystical or magical. They are evidence that it is computational. The unconscious mind is processing information, making connections, testing possibilities, doing exactly the kind of work that neurons were evolved to do. The difference is that it does this work without the overhead of conscious monitoring, without the bottleneck of verbal reasoning, without the limitations of working memory. Your conscious mind can hold, at most, about 4 to 7 items in working memory at any given time. Your unconscious mind is not bound by this limit. It can process vastly more information in parallel, which is why it sometimes finds solutions that the conscious mind, working sequentially through a limited set of possibilities, misses. So the stranger is not just keeping the lights on and the heart beating. The stranger is also your most powerful creative engine. The conscious mind sets the problem. The unconscious mind explores the solution space. And the best outcomes tend to happen when the two systems work in sequence. Conscious effort followed by unconscious incubation, followed by the conscious evaluation of whatever the unconscious produces. This is why sleep matters for creativity as well as for memory. During sleep, the constraints on neural activity are loosened. The prefrontal cortex, the evaluator, the critic, is offline, and the networks of association that link one idea to another are free to explore connections that would be suppressed during waking life. The nightly editing session that the hippocampus performs, replaying and consolidating the day's experiences, also creates opportunities for new connections to form between memories that were previously unrelated. And sometimes, those new connections are the seeds of creative insight. So when you wake up tomorrow morning with a new idea, or a new perspective on a problem, or even just a strange dream that seems to contain some buried meaning, you will know where it came from. The stranger was at work. All of these stories, Gage, the split-brain patients, the sleepwalkers, HM, the improvisers, they share a common thread. In each case, someone lost or gained access to a piece of the brain's machinery, and the change revealed something about how the whole system is organized. And that leads to a question that feels very personal, especially late at night, when the day is behind you and the future is uncertain. Can the brain change? Is the brain you were born with the brain you are stuck with? Because for most of the history of neuroscience, the answer was assumed to be yes. Once your brain matured, once the critical periods of development closed, the structure was fixed. You had what you had. Damage was permanent. Loss was permanent. And then researchers began to discover that the brain is far more changeable than anyone had imagined. Let me tell you about a friend of mine, or rather, a friend of a neuroscientist named Nancy Kanwisher, who teaches at MIT. This friend, whom Kanwisher calls Bob in her retelling, was a brilliant, witty, successful professional. He held a high-powered job. He wrote beautifully. He was the life of every party. But for several years, Kanwisher had noticed something that she could not quite bring herself to confront. Bob kept getting lost. Not in unfamiliar places, but in places he had known for years. Once, a friend asked Bob for directions from his house into Cambridge. Bob said, "You go to the end of the driveway and turn left." His friends looked at each other. "Bob, that is the wrong direction." He thought about it, confirmed his answer, and was wrong. Another time, he could not find his way to the grocery store in his own hometown, a trip he had made hundreds of times. Kanwisher noticed these signs, but she could not make sense of them, because everything else about Bob was completely normal. His intelligence, his language, his personality, his social skills, all of it was intact. How could someone so obviously brilliant be unable to navigate? The answer became clear one night when Bob collapsed at Kanwisher's house. He was rushed to the hospital, and after a long day of tests, the doctors found something in his brain. A mass the size of a lime, sitting deep in the cerebrum, right next to a region called the parahippocampal place area. This region, which Kanwisher's own lab had discovered years earlier, is specifically involved in the recognition of scenes and the processing of spatial information needed for navigation. And then Kanwisher remembered something. Years before, Bob had volunteered as a subject in one of her lab's brain imaging experiments. His scans were still in the files. When they pulled them up, there it was. The same mass, but smaller, the size of a grape. It had been growing slowly for years, gradually pressing on the very brain region that handles navigation, and nobody had noticed it. The tumor was a meningioma, not cancer, but still dangerous at that size. It was surgically removed in an 11-hour operation. Bob recovered beautifully. Within days, he was back to work, sharp as ever, his personality and intellect completely intact. But his navigation never came back. He still cannot find his way in unfamiliar places. He relies on his phone for GPS directions everywhere. He cannot draw a floor plan of his own house. He cannot imagine what is behind him when he is standing in a room. His spatial memory is, as Kanwisher puts it, blitzed. And yet, everything else works perfectly. This case illustrates two things that matter deeply for our story tonight. The first is the extraordinary specificity of the brain. Bob did not just get a little bit less intelligent, he lost one very specific mental ability while retaining every other one. This is what happens when a particular piece of specialized brain machinery is damaged. The rest keeps working. The person is still there, still whole in almost every way, except for this one missing piece. The second is the question of recovery. Kanwisher notes that if all of this had happened to Bob when he was 5 years old, his brain probably would have compensated. Children's brains are enormously plastic, meaning they can reorganize, rewire and redistribute functions to healthy tissue when damage occurs early in development. But adult brains have largely lost this plasticity. The specialized circuits are locked in place, and when they are damaged, the function they support is often lost permanently. This distinction between the young brain and the adult brain is one of the most important facts in all of neuroscience. The brain you have as an adult is in many ways a less flexible organ than the brain you had as a child. The critical periods, the windows of time during which specific circuits are shaped by experience, have largely closed. The wiring has been set. And this is why childhood experiences matter so much for brain development, and why damage early in life can be compensated for in ways that damage later in life cannot. But plasticity is not completely gone in adulthood. It is reduced, but not eliminated. You can still learn new things, form new memories, acquire new skills. The synaptic modifications that underlie learning, the strengthening and weakening of connections, continue throughout life. What you cannot do as easily is rewire the basic architecture, the large-scale organization of which regions do what. That organization was laid down early and is now mostly fixed. And here is one final twist on the question of brain changeability, one that pushes the boundaries of what we thought we knew about inheritance. A neuroscientist named Bianca Jones Marlin studies how experience can change the brain across generations. In her experiments, she took mice and paired a specific smell, the smell of almonds, with a mild electric shock to the feet. The mice learned, as you would expect, to fear the smell of almonds. When they smelled it, they froze, their stress hormones spiked, they showed all the signs of fear. But then Marlin looked at the noses of these mice, at the specific receptor cells that detect the almond odor. And she found that after the fear conditioning, the mice had grown more of these receptor cells. Their noses had physically changed, producing more neurons that were sensitive to the smell that had been paired with danger. It was as if the nose was saying, "This smell is important in this environment. We need to be better at detecting it." And here is the astonishing part. The offspring of these mice that had never been shocked,
that had never experienced the almond shock pairing were born with more almond-detecting receptor cells in their noses than normal mice. The fear memory, or at least the biological preparation for that fear, had been transmitted to the next generation. Through some mechanism involving changes in the sperm cells, the experience of the parent had altered the biology of the offspring.
This is a form of epigenetics, the study of how gene expression can be changed by experience without altering the underlying DNA sequence. The genes are the same, but which genes are turned on or off, how actively they are expressed, can be modified by environmental events and then passed down. The mice did not inherit a memory of almonds and shocks. They inherited a heightened sensitivity to the relevant smell, a kind of biological preparedness that made them quicker to detect and respond to the same stimulus that had been dangerous for their parents. Marlin has reflected on why this research matters beyond the laboratory. She says that as humans, we know how environment and traumatic events change people. We have always known this, intuitively. What the science does is bring that knowledge to an undeniable truth by showing the physical mechanisms through which experience reshapes biology, not just for one lifetime, but potentially for generations. Your brain is not a static thing.
It was built by genes, shaped by experience, modified by learning, and influenced by the experiences of people who lived before you. The stranger in your head carries traces of lives that were lived before yours began. And there is one more thing worth knowing as you lie here. The very act of listening to these words, right now, is activating one of the most extraordinary systems in your brain. When you hear speech, the sound waves enter your ears and are converted to electrical signals by the hair cells of the cochlea in your inner ear. These signals travel along the 8th cranial nerve, the vestibulocochlear nerve, to the brainstem, where initial processing begins. From there, the signals are relayed upward through the midbrain, through the thalamus, and into the primary auditory cortex in the temporal lobe, where specific neurons begin to recognize individual sounds, phonemes, the building blocks of language. But recognizing sounds is just the beginning.
In Wernicke's area, those sounds are assembled into words. In a region that neuroscientists call the associative temporal cortex, those words are matched to meanings. And in the associative parietal cortex, something even more remarkable happens. The meanings of individual words are linked together into complex ideas, connected to visual images, to emotional associations, to memories, to everything else you know about the world. A single word like ocean activates not just the sound pattern and the dictionary definition, but the visual memory of waves, the smell of salt air, the feeling of sand under your feet, the emotional associations of every trip to the beach you have ever taken. The word is a key that unlocks an entire constellation of experiences. This network, the one that connects words to images to emotions to memories, is sometimes called the speech model of the external world. It is built up over years, starting in early childhood, when every new word you learn creates a new node in the network, connected to every other node by threads of association. By the time you are 4 years old, you have a functional vocabulary of several thousand words, and the network they form is a working model of your environment, a simplified but usable representation of everything you have encountered. And this model is not just a passive record, it is an active tool. When you think, when you reason, when you plan, what you are doing, at the neural level, is sending signals through this network, from node to node, following chains of association, testing connections, generating predictions. Thinking is movement through the speech model. And when the movement produces a new connection, a link between two ideas that were not previously associated, you experience that as insight, or creativity, or understanding. Your brain rewards these moments with a small burst of dopamine, the neurotransmitter of pleasure and motivation, because new connections are, from the brain's perspective,
valuable. They expand the model, making it more powerful, more predictive, more useful. Right now, as you listen, your speech model is active. Each word I say is landing in the network, activating associated nodes, building temporary connections, shifting the overall pattern of activity. You are not just hearing words. You are running a simulation, a model of the world being updated in real time by the incoming information. And when you fall asleep, the hippocampus will replay some of these patterns, consolidating the most significant ones, discarding the rest, editing your model in ways that you will never be aware of. This is, perhaps, the most intimate thing the stranger does. It maintains and updates the model that you use to understand the world and your place in it. It does this every night, in the dark, while you are absent. And when you wake up, the model is still there, slightly different from the night before, and you call that difference learning. You call the model understanding. You call the whole process of having a model and using it to navigate the world thinking. And all of it, every bit of it, is the collaboration between the small conscious voice that says, I, and the vast unconscious machinery that makes the voice possible. And now, let us come back to where we started. Right now, as you lie here, your brain is doing all of the things we have talked about tonight, and it is doing them all at once. Your brainstem, that ancient, thumb-sized structure at the base of your skull, is generating each breath you take. The respiratory pacemaker neurons in your medulla and pons are firing roughly once every 5 seconds, sending a signal down to your diaphragm and your intercostal muscles, and your lungs are expanding and contracting in response. You did not ask for this. You do not need to supervise it. It happens on its own, the way it has happened every moment of your life since you took your very first breath. Your vagus nerve, that great wandering cable, is carrying signals from your brainstem down through your chest and into your abdomen, adjusting your heart rate, slowing your digestion, quieting your body's alarm systems. The parasympathetic branch is taking over, telling every organ it reaches that the day is done and it is time to rest. Your thalamus, that walnut-sized relay station deep in the center of your brain, is gradually closing its gates. During the day, the thalamus passes sensory information up to the cortex, keeping you aware of the world. But as sleep approaches, the thalamus begins to filter more aggressively, blocking incoming signals, reducing the flow of information to the cortex. The outside world is fading. The sounds in the room, the sensation of the mattress, the temperature of the air, all of these are becoming less vivid, less present, as the thalamus dials them down. Your hippocampus, that seahorse-shaped structure in your temporal lobe, is preparing for its nightly work. Once you fall asleep, it will begin replaying the day's events, consolidating the ones that matter, transferring them to long-term storage in the cortex, and discarding the rest. Whatever you experienced today, whatever conversations you had, whatever you saw and felt and thought about, the hippocampus will sort through it all while you are unconscious, editing your autobiography without your knowledge or consent. Your prefrontal cortex, the decision-maker, the planner, the self-monitor, is winding down. The frontal lobes are among the first regions to reduce their activity as sleep approaches. The interpreter is going quiet. The narrator is putting down the pen, and the rest of the brain, the ancient machinery that was running your life long before consciousness evolved, is taking the night shift. Your amygdala is still on watch, still scanning for threats, still ready to jolt you awake if something dangerous happens. But in the absence of actual danger, it too is settling into a lower level of vigilance. Your cerebellum, that miniature brain at the back of your skull, is maintaining the last vestiges of muscle tone, keeping your body positioned in whatever way you have settled into. It will continue to make small adjustments throughout the night, shifting your weight, turning your head, doing the quiet, physical maintenance that keeps your body functional while you are away. And somewhere in the depths of your brainstem, the sleep centers are releasing their signals, the neurotransmitters that promote sleep, the inhibitory signals that quiet the cortex, the chemical tide that pulls you gently, gradually, toward unconsciousness. The blue spot, or locus ceruleus, that cluster of norepinephrine-producing neurons in the upper pons, is reducing its firing rate. During the day, it keeps you alert, focused, responsive to novelty. As you approach sleep, it goes quiet. The watchman is standing down. All of these systems, every one of them, are operating without your input. The stranger is doing what the stranger has always done, running the ship while the captain sleeps. And here is the thing that I hope you can take from tonight's story as you drift off. The stranger is not a stranger at all. The stranger is you. The unconscious processes, the automatic adjustments, the emotional guidance, the memory consolidation, the motor coordination, the sensory filtering, all of this is you. The conscious narrator, the voice that says, I, is one small part of a much larger system, and that system has been keeping you alive and functional since the moment you were born. The feeling of being a single, unified person is a beautiful and useful construction. It allows you to act in the world, to plan your future, to remember your past, to connect with other people. But underneath that construction, there is something even more remarkable. There are 86 billion neurons, organized into networks of staggering complexity, carrying out computations that no single one of them could perform alone, producing an experience, your experience, that is richer and stranger and more wonderful than any one part of it could ever account for. You are not a single voice, you are a chorus. And the music they make, the music your neurons make together, is the most intricate and mysterious phenomenon in the known universe. The scientists who study the brain, people like Sperry and Gazzaniga, like Broca and Wernicke, like Damasio and Eagleman and Kanwisher and Molaison, they have spent their careers trying to understand that music. They have made enormous progress. They know a great deal about the instruments, the neurons and the sections, the brain regions. They know something about the harmonies, the way different regions work together, and the dissonances, what happens when parts are damaged or disconnected. But how all of it comes together to produce the experience of being you, the feeling of consciousness, the sense that there is someone home behind your eyes, that remains, as one neuroscientist put it, the hardest problem in science. We know where it happens. We know many of the components. We can see the circuits lighting up.
But we cannot yet explain how a 3-pound mass of cells generates the experience of being alive. And maybe that is okay. Maybe that is a mystery worth being comfortable with, because right now, your 3-pound mass of cells is doing something beautiful. It is winding down. It is letting go of the day. It is transitioning from the bright, noisy, effortful state of wakefulness into the quiet, restorative state of sleep. The prefrontal cortex is going dark. The thalamus is closing the gates. The parasympathetic system is taking over. The hippocampus is warming up for its nightly editing session. You do not need to understand any of it. You do not need to supervise any of it. The stranger has it covered. The stranger has always had it covered. Your breathing is slowing down. Your muscles are relaxing. Your thoughts are becoming softer, less defined, drifting like clouds in a quiet sky. The electrical storms of the day are fading into the slow, deep waves of sleep.
Everything we talked about tonight, the neurons, the synapses, the hemispheres, the memory systems, the emotional circuits, the illusion of unity, all of it is working right now, inside you, doing exactly what it evolved to do. Billions of cells, trillions of connections, all of them conspiring to produce this moment, this quiet, this letting go. The stranger in your head is not a stranger. The stranger is the rest of you. The parts you never meet, the parts that work in the dark, the parts that have been keeping you alive and dreaming and learning and feeling since before you could speak. And right now, all of those parts are saying the same thing. Rest. Just rest. The brain will still be here in the morning. The mystery will still be here. The chorus will still be singing. But for now, just let the music fade. Let your eyes stay closed. Let your body be heavy. And let the stranger do what the stranger does best. Carry you, gently, reliably, into sleep.